excited to be here with y'all this afternoon. I think it's a really exciting way to kick off ASGCT's annual meeting with this workshop that's trying to bring together, where do patient advocates fit into the overall drug development pipeline? How can y'all drive your science forward? So we have a two-part session set up for you guys today. But first, some reminders and housekeeping for the class. So this workshop does require additional registration fee to attend. So if the back of your badge does not already have this workshop listed, please do take a moment to either register online at your seat or go over to the registration tables and they can help you with that as well. And then all annual meeting attendees are expected to be respectful and conduct themselves in accordance to the meeting code of conduct. I don't anticipate that to be an issue. Then attendees are prohibited from using any flash photography. So please do your best to not distract your fellow workshop attendees or our presenters or panelists who are up here and donating their time and expertise for us. And then finally, the Wi-Fi network is MCEC Wireless Network. There's no password required. And then you can also download the ASGCT Events meeting app for free wherever you download apps for your phone. Okay.
And thanks again, everyone, for joining the workshop today that's entitled Shared Decisions, Shared Success, Engaging Stakeholders in Cell and Gene Therapy Translational Research, which is co-hosted with the National Organization for Rare Disorders. So we very much appreciate their partnership here. Our first speaker will be Dr. Allison Bradbury, who serves as a principal investigator and associate professor at Nationwide Children's Hospital. She will be presenting on building a strong research program and early investigator engagement. After Dr. Bradbury speaks, we will also have Ms. Rhonda Rowland, who is president of the Wilson Disease Association. She will be sharing her experience on engaging the FDA, preparing for a patient-focused drug development meeting. Dr. Bradbury.
All right, thank you to Sam and Kim for inviting me to speak today. So as was mentioned, I'm a principal investigator at Nationwide Children's Hospital in the Center for Gene Therapy and hold dual faculty at Ohio State and Department of Pediatrics. And I also sit on the board of directors for ASGCT. And today I really just wanted to kind of talk about how we can build strong research programs and really that relationship between PAGs and academic researchers and industry. So I know this is the very beginning of the meeting, so just stick with me, but we're gonna go all the way back just briefly to the central dogma of biology. So what we all know is that we have our DNA, and our DNA, our DNA is made into RNA, and our RNA is made into this amino acid chain, which makes our functional units of our protein. This amino acid chain is read in an all-important three-letter code, or the codon, and depending on the order of those three-letter codes, determines which next amino acid comes in the amino acid chain, which is the instruction manual for your proteins. If we have just one change in our genome, we have over 3 billion letters that make up our genome, whether this is an insertion, a deletion, or a misspelling, this is what results in dysfunction of our proteins. So our proteins could be completely degraded, or they might be shortened, or they might be changed in their structure and function. And so here what we know is that we have genetic changes that result in a dysfunction of a protein, and this is what is causing disease. And now what we all want to know is if we have this genetic change that is resulting in a disease, can we fix the gene defect? And if so, what's the best tool? So we have some molecular changes that result in the loss of function of a protein. So the protein is short or absent or degraded. And so a therapeutic strategy for this might be a gene addition, where you're trying to provide a healthy functional copy of a gene to compensate for the loss of function. You might also have a therapeutic strategy of an antisense oligonucleotide. These might be especially beneficial for what we call splice site defects, where the introns and exons have a junction where there's a mutation right in that junction. So in this instance, an antisense oligonucleotide might be your best therapeutic tool. However, there are other molecular changes that result in accumulation of a protein, and these are called gain of function. And this accumulation of a protein results in toxicity. And these indications take a completely different therapeutic strategy. So they would require a gene knockdown, where you'd want to reduce the amount of that toxic protein. You wouldn't want to use the strategies where you're trying to make more of the protein, because that would just make your problem worse. And then regardless of if you have a gain or loss of function, we have another genetic tool called gene editing, where you use little molecular scissors that can change defects, very specific defects, and these are usually more common for individualized or precision medicine. So if we know that we have a change in our genetic code that's resulting in a disease, and we have a number of different molecular tools, and we're really interested in, okay, can we use one of these tools to attempt to fix our disease? And it sounds easy, but there really is quite a long roadmap from really disease characterization to therapy development. So first, we really need to understand the disease mechanism. And you really cannot develop a therapy if you do not comprehensively understand the disease mechanism. You could end up targeting the wrong cell types or pathways and end up doing more harm than good. Also, in addition to really understanding the disease mechanisms, you need model systems to exist. Whether they're in vitro, in a dish, or in vivo, you cannot evaluate therapeutic efficacy without the appropriate model systems. Once you have a complete understanding of your disease mechanism and you have generated model systems, then it might be time for that therapy development that we just showed, the different therapeutic tools. And once you've decided on your therapeutic tool, then you use your model systems to evaluate that therapy, and we usually do efficacy and dosing studies. If at that point you're able to demonstrate benefit in your model systems, then we proceed to safety and tolerability studies. And finally, at that point, we hope all of this really ends up with human clinical trials. And what I would say for the PAGs is that really at each of these steps, you need to reevaluate who is your strategic partner, and then at each step also evaluate, are we making the right strides? Are we meeting milestones? And also, is there a different or another strategic partner that could help for the next step? This roadmap is also going to look different for each treatment modality. For example, if you're trying to develop an antisense oligonucleotide, you might not need an in vivo model system. It also differs for each disease because you're going to have a different starting point. Some diseases, there's model systems that already exist and are well characterized, and for others, there are not. So it really is going to depend on the starting point of your disease. So I'm going to start with the very first point that I made, and that's in vitro model systems. And there's really been big strides and advancements of in vitro model systems over the last decade, and I think this is a pretty easy, low-hanging fruit for PAG engagement. So we used to rely a lot on these very simple 2D cultures such as fibroblasts, but now with advancements, we can make these 3D organoid models, which are little organs in a dish. And these are really pretty simple. So they come from just patient blood sample or patient fibroblasts, and these blood samples, we take out the peripheral blood mononuclear cells, convert them to induced pluripotent stem cells, and from those stem cells, we can derive almost any cell types. You can make liver cells or lung cells or brain cells, and then from those you can continue on, if you choose, to make these 3D organoid models. And so these are really helpful in vitro model systems that can help to understand the mechanism of disease. And there are also organizations now that can help PAGs and researchers with this step. So again, this is ways to try to move things more efficiently and try to also take off some of the work from academic labs if needed. And so one example that I've worked with is Simon's Searchlight. And so if your gene is a Simon Searchlight gene, what they'll actually do is they'll collect the patient blood sample. So if you have a patient conference, they'll even come and bring a phlebotomist and do the blood collection at your conference. Otherwise, your patients can go to the Quest lab, and then they send the blood to the Simons Foundation, and the New York Stem Cell Foundation gets it all the way to those iPSC stage, and then they basically give them. They sell them to researchers for under $200, which is really ridiculously cheap because it costs us thousands of dollars in months to get to that step. And so even as Simon says, these blood cells can be used for disease modeling, which is really important, but they can also be used for drug screening and discovery. So I think this is one kind of easy way and low-hanging fruit that PAGs can really help drive research. In addition to in vitro model systems, a lot of times we unfortunately do need to make in vivo model systems, and this can be complicated. There's a number of different types of in vivo model systems. We tend to use mice because they're easy to genetically manipulate, and they're relatively cheap and easy to breed. But there's a number of different types of mouse models that you can make. Traditionally, what was used was these knockout mouse models where you knock out the entire gene. But unfortunately, this makes none of the protein, and so a lot of proteins, when you have none of that protein, it's not compatible with life. So knockout mouse models really aren't that great of a model. Even if they are viable, they often don't really recapitulate the disease. So you could use inducible or conditional mouse models to turn on or off genes at different times or in different cell types. You could use knock-in mouse models where you put in a specific patient mutation. Or sometimes we need to use humanized mouse models, particularly if we're doing something like gene editing, where we need to edit a specific mutation in a human that might not be conserved in the mouse genome. So another way that PAGs can really help with this step is trying to help researchers identify, is there really a specific type of mouse or variant that's going to best model our disease and be most useful for our treatment. So for example, if your indication has like a mutation hotspot that has a lot of patients represented by that mutation, that might be the best bang for your buck for putting in a knock-in mouse model. But again, it takes a lot of thought into what you're trying to model and what treatment approach you're going to use. So once you have your understanding of your disease mechanism and you have some in vitro and or in vivo models at hand, that's really the time when we start to think about that therapy development and what are our molecular tools. And again, I already went through these, but our toolbox is growing constantly and shaping. I mean, even with gene editing, now we have base editing and prime editing, and the delivery modalities to deliver these, whether they're viral vehicles or lipid nanoparticles, it's an ever-evolving landscape. So these molecular tools that are in our toolbox, it's really a fun time to be in science. But what you're gonna strategize for your molecular tool is also another decision point for a PAG. You might not want to put all your eggs in one basket. You might not want to say, okay, we're just going to do ASOs, or we're just going to do gene editing, or we're just going to do gene therapy. I get that could be scary because you're putting all your eggs in one basket, and if it fails, it fails. But also, if you diversify your revenue too much and dilute your efforts and you try everything under the sun, then that might also slow things down because you're really diluting your resources that you have if you're trying too many things. So it really is going to take a strategic partner and really intentional thought about which of these molecular tools makes the most sense for your indication and where do you really want to invest those hard-earned resources. After you have developed your kind of preclinical study and done those efficacy studies, the next step is the safety and tolerability studies. And what I will say about these is they are really not sexy and they're hard to fund. Tolerability studies are really not fun, but they are necessary, and it's really important to make sure that we're doing really rigorous science and first making sure we do no harm. So what we have to do is test our therapy in healthy animals to make sure there's no unintended consequences in those animals. And these safety studies, they're going to vary widely depending on what your treatment modality is. So for AAV versus gene editing versus antisense oligonucleotides, there's going to be different safety studies that are required. And also depending on the precedent of that therapy. So if you're using, for example, an AAV capsid like AAV9 that's already been in over 8,000 kids and is FDA-approved products, the FDA is going to have more comfort in the human safety data that they already have versus something like a novel capsid that's never been in human. The FDA would likely require something like non-human primate tox studies. And this is really a conversation that we have with the FDA. You know, we have our pre-IND meeting, and we asked the FDA for guidance on our proposed safety and tolerability studies. So this isn't just an investigator, making up their mind by themselves, but it's really a conversation and an engagement with the regulatory body. As far as your clinical trial sites, sometimes it's like really obvious because you have one principal investigator that's your clinical champion that's driving your research, and that's the obvious site where you're going to do your clinical trial. But sometimes it's less obvious because you have multiple natural history sites, or you have multiple people working on this indication. But what I would say for gene therapy, it's really nuanced in the way that you deliver it and monitor it. So it's not something that should just be taken lightly and delivered, at a center that's not used to delivering gene therapies, because there can be some really unique events that occur after gene therapy that can require intervention. And there's also long-term monitoring. So there are gene therapy centers for excellence, and there's also, of course, disease experts and clinical trialists that all need to be taken into consideration when choosing your clinical trial site. But I also want to acknowledge that it needs to be accessible. I know it's a large burden for families and patients to have to travel, not only for the gene therapy itself, but then for the long-term follow-up of 5 years, and to continue to make the journey back to that clinical trial site. So it is definitely a balance of choosing somewhere that has the experience, but also the accessibility. For the FDA, a lot of people, I think, are scared of the FDA, but I really want to say that they are, go back a slide. They're really there to work with us, and there's multiple points throughout the pathway of preclinical, clinical trial, and even all the way to post-marketing that you engage with the FDA. And they really are there to help provide guidance. So one of the key times that we meet with them is this pre-IND meeting, where we say, this is the data we have so far in our cells and our mouse, and this is what we think we want to do with our safety studies. And the FDA really will come back and give guidance about, we think you should look at the animals for longer, or have more animals, or a different model system. So it really is a conversation with the FDA working with us, not against us. And I highly recommend sticking with as many interactions with the FDA as possible, because I've known of organizations who've skipped the pre-IND, submitted the IND, and the FDA didn't accept their tox package. And so then you've really lost time and resources having to go back and repeat those critical studies. The FDA is also really trying to listen more to the patient voice. And so I think this is another place that PAGs can be really involved, is trying to set up these patient listening sessions and these patient-focused drug development meetings, so the PAGs can really work to get their community together to let the FDA hear what's meaningful for your community. I'm going to talk now about what we've kind of thought of as the traditional path of development of these genetic therapies. Traditionally, what would happen is a foundation would come and support an academic partner. That academic partner would then look for other funding, used to be largely NIH, and then at some point we would have convincing data, and we would go to an industry partner, and we would say, please license this program and try to get it into the clinic. And then eventually, of course, the way we really want the most access to a medication is through an FDA-approved product. And there have been many success stories that have kind of gone through this most traditional path. However, especially for rare diseases now, there's been this challenge, kind of unintended challenges of bringing cutting-edge science from bench to market. This is a paper that was published maybe about 2 years ago now. And I don't intend for you to be able to read all of these, but these are all programs, gene therapy programs, that were dropped after they were licensed. So a company licensed them, and then for some point in their development, they said, this indication's too small, this drug is too expensive, it's a one-time treatment with all three of those factors, how are we going to make any money? And they ended up dropping the program. And so this is really challenging for everyone involved, for the PAG, for the researcher, when you've worked for years trying to advance a program, to then have it dropped. So we've seen some changes. Most recently, in December of 25, the FDA approved the first gene therapy from a nonprofit organization. So it'll be interesting to see over the coming years how this really plays out. pricing and access questions I don't think have been fully determined at this point. Then there have been other indications. This is one example of Test Research Foundation, who partnered with UT Southwestern, and then they licensed their product to Taysha. And then they recently gained FDA clearance to launch their first human clinical trial. So this is kind of more of that traditional path. While you have other foundations, many of you in the room might know Terry, and what he did instead of licensing it is he started Alpida Therapeutics, which is a not-for-profit venture philanthropy, and ended up funding a gene therapy trial for his child's indication, SPG50. There are other foundations that are going even earlier in this pathway of development and actually getting involved at the lab space, and in this indication even built a lab incubator to help drive some of the lab-based research. So the role of PAGs can really be at all and any point in this drug development. I also feel like I need to point out the role of CROs, CDMOs, and nonprofit biomedical research institutes, because these can really help drive research. There's only so much academic labs can do with our resources. So sometimes it's good to partner with a CRO. For example, this could be a great opportunity for a tox study to happen at our CRO. I'm not advocating for any of these specific companies, but just giving some examples of Charles River. You need people to drive the research, and typically that was academics, but now we even have examples of foundations such as Fox G1 partnering directly with Charles River to advance their program. And then we also have the JAX laboratory now has a rare disease translational center that many foundations are partnering with. And some of these CROs, they can even do your model characterization for you. They can give your gene therapy, follow the mice, and then even do your tox studies. And so they can kind of be a part of any or all of that pathway of drug development. So these are ways to just provide some more resources and people if academic labs are struggling with those resources. I do want to acknowledge that a lot of people are thinking about these barriers to gene therapy and access to them, and I do want to applaud ASGCT. They had a working group, and they even published a paper after that about overcoming these barriers, and now they have even developed an accelerator program. So we hope that these are some pathways to help improve ways in which we can really move these pre-viable programs. And I'll also say that clinical trials are not possible without registries and natural history studies, and this is a key role of PAGs. We really need to know who the patients are and where they are, and we need to know the natural history study and the benchmarks that we're trying to improve upon. Okay, I'm gonna go quickly through two real-world examples in my lab of one is the traditional pathway, and then I'll just hint at the second one. So one of the first programs that I worked on in getting to the clinic is this disease called Krabbe disease. And really this disease was so poised well to make it to the clinic because it was identified over 100 years ago. We knew the mechanism of disease. We knew the Gauci enzyme was deficient, the psychosine accumulated in the cells, and we even had 3 models. We had a dog, a mouse, and a non-human primate. And starting in the early 2000s, there was an evaluation of these therapies in these animal models. So this disease was really just well positioned because of this 100-year history. There was a standard of care for Krabbe hematopoietic stem cell transplant. And what we knew about transplant is from over a decade of research that it improved the probability of survival if the babies were transplanted early. It improved their neurodevelopmental outcomes. However, the transplant did not improve their fine or gross motor. So we believe that the transplant did not efficiently target the peripheral nervous system, which is really critical in this disease. So what we decided to do for our first clinical trial is we didn't want to take away, let's see if this will play. Okay. We didn't want to take away the standard of care because we knew it had some benefit, and that was a really difficult decision. So we decided to add on an intravenous, a systemic delivery of, I don't know if this is going to play. Can you play it in the back or not? It just should play on the click. Okay, maybe it's going now. Okay, there we go. So what we were doing is adding on a systemic delivery of adeno-associated virus, or AAV, gene therapy. So when we're doing this in the lab, we decide on which of the serotypes or the shell or the capsid that we're using. In this case, we used AAV-RH10 because we know it does cross the blood-brain barrier. And we put in the human GALC gene. Again, we deliver this intravenous or systemically because we wanted to target that peripheral nervous system that the transplant wasn't targeting, but we also wanted faster and better correction to the central nervous system as well because the transplant takes a long time. We know this serotype crosses the blood-brain barrier, and then the virus gets into the cells of interest. The capsid is rapidly degraded, but then it deposits that gene of interest, and that cell now becomes a factory and starts to produce the therapeutic enzyme, so in this case, the GALC enzyme. We do know that we need these cells to be present, so now this cell is acting as a factory and making that really important GALC enzyme. And lysosomal storage diseases have an added advantage that it can cross-correct and get into neighboring cells. But especially for neurodegenerative diseases, as the cells start to die off, we no longer have those cells to act as that factory to receive the virus. So we do know we need to intervene early. So we had a mouse model, as I mentioned, a naturally occurring mouse model of Krabbe. So it was great, it was well characterized, and we tried the combination therapy in the mouse model and showed there was an improvement in survival and there was a dose response. The more gene therapy we gave, the better it worked. At this point, I was fortunate to be at University of Pennsylvania and had a large animal model of Krabbe disease, again, naturally occurring. And this closely mimicked the human disease. So there was tremors, pelvic limb paresis, loss of hearing and vision. And as in the babies, it's really progressive in the dogs, and they reached endpoint by 16 weeks, so we knew relatively early if we were able to really move the needle on this disease. I'm not going into any details of the study because it's published, but we basically repeated what they did in the mouse and the dogs, and we showed that there was a combinatorial effect and that there was a dose response in the dogs. And in our dogs that received the high dose of the gene therapy at 2 years of age were completely neurologically normal. So these dogs had normal MRI and normal nerve conduction velocity and really had a great quality of life 2 years, so significantly extended the survival. So we were able to demonstrate that we corrected central and peripheral nervous system disease in 2 model systems, and we were ready to move this to the clinic. So this was licensed to Forge Biologics, and they dosed 6 kids. We had the added benefit that Krabbe was then added to the newborn screening, so we were catching the babies really early. However, after a year, in January 2005, they made the strategic decision to drop the program. And this was really devastating for the patient community. And it wasn't because of the science. You know, this was a business decision that they really wanted to stay more in the manufacturing space. So more recently, at Nationwide Children's Hospital, we partnered directly with a foundation, SLC 6A1 Connect, and we opted intentionally to not license this program to really retain that control. And we were able to drive this program to the clinic in just about 5 years, just a completely academic nonprofit endeavor. And I'm speaking on this program later in the meeting, so if you want the details on all the steps and the timeline that went into that, please feel free to come to that talk. So I hope that I've given a little bit of balance of hope and reality, because there are so many great advances in our molecular tools that allow us to tackle diseases that we couldn't touch decades ago. However, the road is still expensive and long, and I think the role of PAGs has gotten even greater, and they're involved for a longer duration because we have fewer industry partners that fund this work, and we also have less NIH funding. So the role of PAGs is just really, really instrumental. And I will say that there is some great ASGCT resources. We have a lot of educational resources at this website, videos on what is gene therapy, clinical trials, and we also have a patient press newsletter. So it's really great if you are interested to subscribe to that newsletter, and you're already here for a meeting, so good job on that. And I will not take questions because we'll have a panel, but thank you all for being here today.
Thank you, Dr. Bradbury. We'll also invite now Ms. Rowland up to the podium to give a presentation on the Wilson Disease Association's experience with patient-focused drug development meetings.
Okay, well, thank you. Well, first of all, Allison did a wonderful job, and I appreciate all the kind words about patient advocacy groups and our role in development. And I really appreciate the opportunity to be here today. And in preparing for this, I was thinking back on some of the ironies or coincidences of being invited to speak here today. So I'm from Milwaukee, Wisconsin, that area. And I don't know if you know, but the American Society for Gene and Cell Therapy is also from the Milwaukee area. So yesterday I mapped it, and they are located 12 minutes from where I grew up. So if somebody had told me 50 years ago that I would be here today speaking about on this topic, I certainly would not have believed it. And at that time in my life, I had no idea that I was even living with a rare genetic disease. So I was diagnosed with Wilson disease in 1983. It is a liver disease in which we cannot metabolize dietary copper, so the copper builds up and eventually poisons us to death. And now I'm president of this organization. And I am here, as you know, to talk about what an externally led, patient-focused drug development meeting is. So what is an ELPFDD? First of all, this is a little hard to see. I'll look here. So this is a structured public meeting, and it allows the patient community to directly communicate with the FDA. And there are two areas that we really focus on, and that is the burden with living with the disease, and then also what the treatment needs are. And at the end of doing this, and it's kind of an all-day, you're putting on a show for the FDA. That's better. The findings are then compiled into a voice of the patient report that is submitted to the FDA, and the voice of the patient becomes part of the official public record used in the drug review decisions. And the WDA's decision to pursue an ELPFTD was really based on the fact that we had a number of treatments in the pipeline that we thought it would be beneficial for the FDA to have a better understanding of our disease. And just to kind of go back to that, I mentioned that I was born with this rare genetic liver disease, and when I was diagnosed, there was one treatment available, and it worked for me. And since then, we've had two other treatments developed, and we know we are very fortunate. So we understand from NORD that with their recent scientific meeting, we still have 95% of rare diseases don't even have a single treatment. So we're very fortunate, but as you'll see, our treatments don't work for everyone. We now have two gene therapies that are in clinical trials. We have Ultragenyx in a phase 1/2 trial of UX701, and we also have prime editing. It is a gene editing technique, and we are doing now a pre-interventional trial where they're assessing patient interest. So that's where we are with that. And so that's why we thought it would be a good idea to do one of these. And this is just to kind of give you an overview of the time frame and how long it takes. So I have to say, when we embarked on this, I had no idea it would take this long. And even though I was told it was a lot of work, we really had no idea how much work was involved in it. So for a small patient advocacy organization like ours, it's really a lot to take on. So that's something that a group would really have to consider if they make this decision. So learning from others. So this was critical for us. This here is a photo, goes back to the NORD Summit from 2024 that I attended. And at this time, I had just become the president of the Wilson Disease Association. And in addition to that, I was the acting executive director. We were all volunteers. We had just one part-time paid admin to help us out. So I went to this meeting right after I became president, learned a lot, and one of the things I learned was about the PFDD meetings and the learning sessions. And at that time, I thought, oh, it would be so great if we could at least just do a learning session. You know, that's something I think that our organization could take on and handle. So it turned out, you know, to go a little different direction, but this is kind of what we did to learn more about it. You know, I learned about it first at this meeting. Then what I did is attended any webinar that I heard about that was going to be explaining it, or doing the research and finding out about other organizations and watching their ELPFDDs. And then another part that was very important is I set up one-on-one meetings with other organizations that had done this. And one of the early ones I did was with the Angelman Syndrome Foundation, talking to Ryan Fisher, who was so generous with his time to explain what they went through and putting theirs together. So we used all of this information to help us determine how we would put ours together. So we decided to work with an FDA consulting firm, law firm, HPM, Hyman Phelps, McNamara, and James Valentine in that photo, he actually helped develop this program when he was at the FDA. So he clearly knew the structure and everything that was involved with this. So for us, again, I had mentioned that we were a volunteer organization, a volunteer board. We did not feel that we had the personnel to try to take this on ourselves. And so they had the expertise, so that's why we engaged with them. So fundraising, working with an outside group like this, very expensive. So these were the real costs that we had to plan for. And we were very fortunate because we did have an industry partner that had just come into our community. They wanted to take a drug that was shelved by another industry partner, and they wanted to then take it on and try to get it FDA approved. So they came to us and they said that they would support us in this. And this is what enabled us to move from doing a patient listening session to doing the full ELPFDD. And then from there, we did include all of our industry partners in this, because our goal was to help all the partners and all of the drugs and gene therapies that were in development. So inviting clinical speakers, when you put one of these together, you do have to have an FDA speaker, and we had Dr. Amy Magnarelli from CBER. And then in addition, we needed 2 clinicians, one to do the clinical overview and the other to do the treatment overview. And so we asked our 2 strongest researchers devoted to our disease. And what this did too was lend some credibility to our meeting as we worked on moving forward. The other part that we were told that we really needed was to ensure that we had some endorsers and promoters to help us raise awareness for the meeting and help give us support. And in addition to that, then we worked on our own communications with our community. And this we worked on very intensively for more than a year to try to educate our community. It took me a while to learn what this was all about, and so it was really a concerted effort to communicate to our patients and caregivers. So planning the meeting structure, so we had a virtual broadcast. Some of these ELPFDDs actually take place in person. Some of them are hybrid. Doing them in person is much more expensive. Hybrid is also expensive. And as we all know, since the pandemic, one of the silver linings is we have all become very used to doing these virtual meetings. So community outreach. So this is what we did to try to gather our community together. We needed to have a total of 20 patients participate. And so that included 10 video statements that would be pre-recorded. And then we needed an additional 10 patients or caregivers to be live, as we call it, Hollywood Squares, the day of the ELPFDD meeting, to kind of be so-called conversation starters during the meeting. So the way we did this is we created a survey and sent it out to our community. And we did this in a lot of different ways to try to get as much feedback as possible. And we had more than 50 responses to this. And in the process, we learned a lot about what was really important to our community. And then this is a part where, you know, the fundraising helps. As I mentioned, we had volunteers. I was able to contract with some colleagues. I was a CNN medical correspondent. I worked at CNN from 1986 to 2002. And so I recruited some of my former colleagues who had also worked in health and medical reporting. And so we could have a very unbiased picture of our disease. And so this is us meeting, as we talked about. We would do kind of almost like baseball cards to try to figure out the right picture of our patient community so that we could give the FDA a very broad perspective of what our disease looks like. And planning the meeting structure, some of the additional elements that we had that day. Here you can see what the conversation starters, what that looked like. We also had live polling. We had call-ins, as we say, Larry King style. And we actually kind of got those, some of them set up in advance, so they would be in the queue all ready for this. And then we also took written submissions from our community the day of the event so that they could be coming in. So it was really very multimedia and using a lot of different ways to try to communicate information to the FDA. So living with disease, living with Wilson disease, I wanted to give you an idea of what our videos look like. And one of the things I learned in talking to some people in our planning is you do want to stand out with the FDA. So I worked on the area that I knew best, and that is, you know, to produce videos. And with these were all developed by the patients themselves, that we worked very intensively with them on their statements, helping them to kind of shoot some of their own video with their iPhones and gather photos so we could effectively tell their stories. And you're going to see in this one that we had to use subtitles, and sometimes you need to do that depending on how well the patients can communicate. And in this case, all three of these siblings have Wilson disease, and the two men, they are twins, and both had liver transplants, and so this is their story.
We are Aida, Sergi, and Eugenie. Sergi and Eugenie are 10 years old twins, and Aida is 40. We were born in a town called Cutting Green, Cut Clough. Our childhood was very normal. We went to school, we had excursions. When I was 60 years old, and Sergi and then were 11 years old, Sergi suddenly started to come right very well. Our parents took Sergi to the hospital, and in about 2 weeks, he got the Wilson disease diagnosis. Then the doctors done the test and confirmed Abdeni and me also had Wilson disease. Abdeni and me did not have any symptoms at this time. However, my doctor discovered that he had sugar. We detected in the vein and Jenny had disease. I carried very good in the liver. I took doing like marketing for and then took it for about 2 weeks. And after it's been almost 2 years. We have done that which is comes and comes from the bodies, which not control, of it, talk it, I could
When I started the pursuing, I began to have difficulty walking and talking, but it was not nearly as bad that I was in the
finally our letters did amazing regime and our things into that into space slowly. During this time our parents and grandparents helped us in their lives and take care of us. This is work of us is him we need to work again. Again, in the mouth, again, it again.
When I was 17, and the work references, our father died. Then realized that our mother and grandparents, one day in the future, will also die. So it was important that the three of us become as independent as possible, both physically and financially. I went on to Barcelona to study computer engineering. Later studied computer tech and studied programming programming.
We also started children as part of it, but it's for our education, in dance, sing, and cognition. We were stable for our work until I was 27 years old. We were finished because of the studies and things within the conclusion was 84 years old, just needed the fund. And we were people, the parents,
Now I'm very young, work with computers and study works as a student tutor. I'm briefly apart with my boyfriend Alberto, and we took our care of each other. We try to be active and sometimes take hikes in the mountains.
but these symptoms, challenges, as every we work with difficulty, accidents, calm down. With this, which related to the solution, however,
Psychologically, we feel vulnerability and sometimes fear, because our lives depend on medication.
Hi,
So then the other areas mentioned is on treatment. And for this one, this was again showing where the unmet needs are and where we need additional.
my name is
options.
Kim, and I'm from West Palm Beach, Florida. My son James was diagnosed with Wilson's disease in January of 2020, just months before his 21st birthday. Looking back, his symptoms had begun a year earlier. When he came home from college, he had what we called a wiggle. When he spoke, his head would move. At first, he could control it until he couldn't. We went from doctor to doctor. Our general practitioner had no explanation, and a neurologist ordered a CT scan that came back normal. We were told it might be stress-related to school and changing majors. Blood work showed elevated liver enzymes, but that was dismissed as a virus. Over time, James began to drool, move more slowly, and lose weight because eating had become so difficult. One day at lunch with my husband, James suddenly couldn't chew his food. We admitted him to the hospital with a long list of unexplained symptoms. After 2 weeks, he was misdiagnosed with early onset Parkinson's. It was only because a kidney doctor noticed something in his chart and suggested Wilson's disease that James was transferred to the University of Miami. There he was correctly diagnosed within 3 days. We withdrew James from college to focus on his health. At first, he was prescribed Trientine, and we were told he had a good chance of recovery. But instead of improving, within 3 months, he went from being an independent young man to being bedridden. He lost his ability to walk unassisted, required a stomach tube for feeding, and struggled to speak. This was a very difficult time. It was a lot on our family. His decline was so rapid that Trientine was stopped and replaced with zinc, but things only worsened. He slept all day and was awake all night. This went on for weeks and affected everyone in our home. He also suffered severe dystonia that caused painful contractions in his arms and legs. Eventually, a local neurologist prescribed penicillamine, but the disease continued to progress. Almost 6 years later, our small family's life revolves around James, and we are his only caregivers. He relies on us for everything. Every day begins early with his first medication, penicillamine, which must be taken on an empty stomach. An hour later, we give him a mix of meds, baclofen, clonazepam, glycopyrrolate, and Ingrezza. An hour later, he gets breakfast, which is a carton of liquid food given to him through his PEG tube in his stomach. At 11 a.m., he gets zinc. At 12 p.m., more food and medications. He receives baclofen, clonazepam, glycopyrrolate, and escitalopram. At 3 p.m., more penicillamine, and at 5, zinc. At 6 p.m., food and baclofen, clonazepam, glycopyrrolate. At 8:30, he receives another cocktail of medications, baclofen, clonazepam, glycopyrrolate, risperidone, and a pill to alleviate stomach pain. He has one more meal at 9:30 and then off to sleep. These drugs serve different purposes, some to ease anxiety, others to calm the dystonia, and others to help him sleep. The dystonia shapes his whole body. His right arm is in constant motion, always moving, while his left arm stays curled tightly, his fist clenched. His left leg curls too, while his right leg can extend. It's exhausting for him and for us. Some days, the meds help him relax enough to sleep. Other days, even after his full bedtime cocktail, he's wide awake into the early hours. Some days he's unusually drowsy. Other days, he's what we call extra wiggly. There's no rhyme or reason. It's the roll of the dice, despite us following the same routine every day. Without his medications, the situation is unmanageable. During hospital stays, when doses were delayed or withheld, he would thrash so violently that he once pulled out his stomach tube. He banged his arms and legs against the bed and even threw himself onto the floor at night, bruising himself. Since then, we've fought to make sure he never misses his regime. Someone must always be there with him as well. He can't yell if he needs help. On good days, he jokes, laughs, and teases his sister or my husband, still finding humor where he can. But he also endures constant movement, fatigue, and the uncertainty of what the next hour will bring. What I fear most is the future. What happens when we're gone? Who will care for him? And will they treat him with dignity? Will he be respected, loved, and not left alone? These questions haunt me. My hope and prayer is that new treatments will be developed specifically for neurological Wilson's disease. something that will allow James and others like him to have a better quality of life. Because right now, this disease has taken over not just his life, but ours as well.
So on the day of our event, I remember hoping that we would have at least 100 people show up, online for our meeting. And I remember it was like 15 minutes before it was actually starting, and we worked with Dudley Digital video studio, television studio on this. And he said, well, you already have 50 people who are waiting to get in. And we ended up having 350 people show up for it, which we were so pleased with and very grateful for. And also, it was the same with for the FDA. We hoped that some would show up, and we had 40 FDA regulators who joined our meeting that day. And a few weeks later, when we had our debriefing with the FDA, that was designed to get feedback on our meeting, to help us with going forward with our Voice of the Patient meeting. We were told that there probably would be 3 that would come, and we had 8. And I was there with my pencil and paper, all ready to take notes, and they were just really positive. They were very positive on what they had learned and come to understand about our disease. And so I felt very proud of our community for how they came together to help put this on. So immediately after the live meeting, it went on our website, on YouTube at that point. Voice of the Patient report, this is where we are right now. This is taking everything from that meeting, condensing it down to less than 30 pages. And the science writer that we engaged for this, she said this has probably been her most challenging one to do, because our disease can look like a liver disease, neurological, psychiatric, or a combination of all of those disorders. And that's why Dr. Wilson, who discovered it, had called it the Great Masquerader. So we now have reviewers who are going through it, and once we're done, then we will send it to the FDA. They'll take a couple weeks to review it, and once we're all happy with it, it will get posted publicly. And here's just kind of a graphic to show you how that takes place. How the FDA uses the Voice of the Patient report, it informs the drug reviewers about patient experiences and the unmet medical needs. And when I'd gone to one of the NORD summits, there was someone there, another patient advocacy organization, and at their booth, they showed how one of their comments was cited in the FDA approval of one of their drugs. So that was something that we tried to encourage our community with, that this should make a difference. And so again, that was the timeline. What I didn't talk about, we did a community webinar to try to educate our community about what the big day would look like and how they could participate. And what worked well, kind of have that outlined there. Doing differently, we were just kind of trying to get through everything with our staff. And I would just say that we are so focused on the big event that I would say it's very important to come up with a plan for afterwards. And that's where we are right now, and what to do with the report and getting it to the FDA reviewers who attended our meeting. So thank you all. Here are some resources, and if any other PEGs need some help, I'd love to pay it forward and be happy to speak to any of you. Thank you.
All right. What wonderful talks we had. And Rhonda, if you want to actually hop up here. Yeah, we'll just invite Dr. Bradbury back up, as well as our 2 other panelists. So the next section, we're going to get to move into a Q&A section, a panel discussion, collaborating with stakeholders, and really the goal is, how can we align those therapeutic strategies, shared decision-making, and de-risking. And so we've got 2 additional panelists up here with us. So we've got Dr. Matt Fuller. So he's a vice president and head of gene therapy research at Ultragenyx. And then Jennifer Millian. So she is vice president of early stage programs at Insmed Incorporated. So thank you guys so much for joining us.
Thank you.
So now we are going to welcome our attendees to ask questions of our presenters and our panelists. So if anyone in the room has questions, I invite you guys to come up to the microphones that are in the aisle, and we'll work through those questions. But maybe to get us kicked off with one. So now we've got a couple of industry reps up here, so we've got nice perspectives from academia, patient advocacy, as well as our industry partners. So if we're thinking about those early collaborations in those early stages, industry often has assumptions about what patient advocacy groups need to bring to the table in those early days. But the reality doesn't always meet their expectations on either side. So Matt and Jennifer, bringing in that industry perspective now, what do you think was most helpful as patient advocacy groups first contacted you? What was most valuable that they were able to bring to the table? And then maybe from the other perspective, from Rhonda and Allison, what surprised you most as you were starting some of those industry partnerships? And what were some of those shocking pieces, like, oh, I wasn't expecting that piece? So maybe we'll start with Jennifer.
Sure. So in my career, thank you. So in my career, I've mainly worked in rare disease, and in rare disease, there's often no data or very little data. And I think from patient advocacies, I started in the lab, and I always thought, okay, what we need is data, data, data. And that is true. But what I've actually seen kind of move the needle, particularly in my career, has been the in-life experience. And so the patient advocacies hold that data. They hold the everyday struggles. They hold the progression, and they truly understand how the disease manifests. And I actually have one example that I'd like to share. Working in pediatric neurology, we were talking to a mom, a caregiver at one point, and she was just talking about the fatigue that happens on a daily basis with her child. And we were in the middle of designing a clinical trial at the time, and we had a battery of assessments that we were going to run in the clinic. And hearing from that mom about the fatigue, we just kind of took a step back and said, we can't do a full day of assessments with a child and expect to have really strong data. And not only that, we can't do that to a child that has extreme fatigue. And so I think sharing stories, getting that data, that everyday data out, not only in the aspect of natural history studies and registration data that we talk about so often, but also just really sharing the lived experience of the disease, it really makes a meaningful change. And I think that as folks in industry, we also have to listen, and we have to invite patient advocacy groups to the table to be part of that conversation, because there's so much learnings that you can get just by having conversations. And to me, that's really, really important. But also on the data side, I'll just add one more thing. I've been working on designing a natural history study for the past couple of years, and really input in data that is available or out there. Even if you start in an Excel file, just collecting progression of data, just get a start, get the data out there, and really start trying to put it together. So that was our starting point where we started in designing our trial. So anything you can do to kind of pull data together is really, really important.
So I can't add much to that. That was a very thorough
Sorry.
answer. No, that was a great answer. Maybe I'll just add two things. One, to pile on about the patient experience. We can talk to a couple doctors, we can read a review. That doesn't always tell us everything that's known about that disease, and that's where you guys come in, because you know the disease in and out, and more importantly, you know the homo and heterogeneity of that disease. And that helps us not only design a drug, but it helps us understand what we need to look for clinical efficacy. Because if you don't understand the disease at multiple levels, as was explained for Wilson disease, you can very easily look at the wrong endpoint and not know until it's too late. I think the other thing I would add is you, more so than anyone else, probably knows where the patients are. And that's important for two reasons. One is obviously we want to get the drug to those patients, but two, in order for you to de-risk this for industry, they need to know how many patients there are. And if you go to Gene Review or anywhere and look at diseases, they often just have this big, like, 1 to something number, 1 to 150,000 or 250,000. If you actually know that that is true or not and where those patients reside, that is incredibly helpful.
Yeah, I'll kind of double down on something that Matt was just saying about really helping with the drug development. So one thing I would like to add is like, what is a clinically meaningful outcome when we're talking about drug development? Because as mentioned, a lot of these indications, they are very heterogeneous, and different families might find something else clinically meaningful, and it really helps when we're trying to develop a drug to know what is clinically meaningful, especially when we start talking about genetic epilepsies or neurodevelopmental diseases. A lot of times these therapies are not going to be a complete home run. You know, we cannot reverse disease. So as we're trying to strategize when to intervene and, you know, what our patient population looks like, knowing what is clinically meaningful for the patients and their families, because I think sometimes there are outcomes that the families would really like to see and that would be meaningful for them that a clinician might not find as meaningful. So that's really helpful. And then the other thing that Matt was just saying about we need to know where the patients are and what the natural history study is, and that's absolutely key. Like, we cannot go to the FDA to try to start a clinical trial if we don't know who and where the patients are and what the disease baseline is. And I feel like there's a lot more tools now with registries and even online natural history studies where groups can start to even do things like Bailey's or Vineland's and start to collect them and collate them online. Of course, in-person natural history study is often needed, but at least trying to figure out what those outcome measures are and get that baseline data is really important.
What I would just add to that is, because we do have a patient registry, the Wilson Disease Association does, but I don't think that captures like what we really learned from our ELP-FDD, and I even learned from it, what the treatment burden truly is for these patients, because they have to take their medications away from food and then avoid foods with a lot of copper, and how much of a burden that is. And I don't know that a registry really captures that kind of information. And, you know, I feel like all of our industry partners really took in everything from our meeting and really could see what the patient needs were. And I was more surprised, really, that our doctors were not all watching it, because I thought they're seeing them, the patients in the clinic, and they may be seeing something, but they're not really seeing the full picture. So that, for me, was probably the biggest surprise. But again, very impressed that our industry partners were so engaged with it. And a question I would have probably for Matt, because our industry partners have approached us to create ad boards very early in the process, which I think is really useful to meet with groups of patients. But how that information differs, or was it maybe supplemented by watching the ELP-FDD?
I think what we do is put all of that information together, because what we're trying to do is present a really clear story to regulatory officials that this is needed and it's working. And I think, as someone said earlier, the clinical presentation can be different. The endpoints might be a little bit different, and the benefit a patient sees might be a little bit different from person to person. But if we can present a cohesive story of why this is important and how it's helping, that helps both of us.
So can you further describe, Rhonda, like, when is the time that PAGs should embark on this? And then maybe Matt, like a follow-up, like, if as you're making that complete package, like, what are the next steps? What comes after that to make that package richer, and kind of like the order and the timeline? Like, when should a PAG really invest those resources that you just explained that took that year?
All right, so the resources for the ELP-FDD,
Yeah.
that part.
When should they embark on that journey?
I think you do when you really think that there's going to be a viable treatment that could come along. When you know that some industry stakeholders are going to go to the FDA to see about doing a trial so that they have an idea about that disease. You know, I talked about my CNN days, and I did report on FDA advisory hearings, and that was for big diseases that everybody knew about. So they don't know about our diseases. So to give us the best shot at progressing, I think you need to do it when there actually is some interest.
What was the question that you had asked me?
Because you were just saying that you put that kind of all together as like a package. So then, like, after the PFDD, like next steps, or when should PAGs like engage on different things to set you up for the most success?
I would argue that it's good to engage early and often because we're trying to understand the disease. I think everyone up here knows that there's at least one thing about a rare disease, each one that's probably untrue, and it might take us years to find that out. And so the more we're talking, the more likely it is we find out what's really, really true and what really isn't. The other thing I'd like to highlight, just as an aside, is sometimes I talk to people running PAGs and they're like, oh, look at this person that has this skill set, or this person's a doctor, they're going to do really well. I've yet to meet someone that was on CNN and worked for CNN. But what's important, right, is you took your skill set and you applied it in a way that is so helpful to the patients that need you. And I would just invite everyone that's working with a PAG to think about what it is you have to offer, and then bring people in that have other skill sets.
I really believe in the 80-20 principle there. And when I became president, looking at our board, I gave them all director titles in their area where they had a skill set or a passion or an interest. So yes, I think that really is helpful in this space.
Can I just add a comment about timing? So I work in research, and so we're very much so before the clinic. But we actually ran an ad board last year where we had healthcare professionals, we had doctors, patient advocacy group members, as well as research biologists. And I think it was not too early at all, because it was fun, the conversations that were ongoing where you had the clinician listening to the patient's story and actually the patient's borderline challenging some assumptions about the disease that have been fundamental for years right there in the room, and then the research biologist connecting the dots and tying it to their animal model or the mechanism of action or the disease biology right there in the room. And I actually think it can drive decisions earlier by having that engagement before you even have maybe a drug development program, because the fundamental understanding of the way clinicians look at things, the way patients look at things, and the way research look at things, honestly, is a totally different lens. And with industry in the room as a collaborator, it is fundamental to getting a stronger program, in my opinion. So just wanted to add that.
That kind of brings us into kind of a thought that we were having before about, Allison, you presented kind of that traditional path, kind of starting of drug development, starting in the academic realm, moving into industry, really early on the advocacy representatives, the patient population engaging with academia, and then maybe moving into a biotech or pharma space. But it seems like there can be tension at the time of that handoff from partnering with an academic to moving into a partnership with a biotech company. And I wonder, maybe Allison or Rhonda or even others in the room, if you could maybe speak to considerations as you kind of move through that process, the traditional process, and engaging from that more research-driven academic partnership to one that involves a commercial partner.
Yeah, I think one of the biggest things is the quality and the documentation of your work. To make something that can go into an IND is just a completely different level than just running, basic science research. So if you are going to be conducting a preclinical study that is going to be part of investigational new drug application, the level of documentation and rigor of your science is just at a different level than if you're doing something for exploratory reasons. So I think really kind of knowing which studies are meant for which and having those well-defined. So if this is an IND-enabling study, going into it with that knowledge and making sure everything is being documented, paper-trailed, everything that you would need so that when you hand it off to an industry partner, it's as buttoned up and as tidy as possible. Because if they have to come back or they can't find a chain of custody of a virus that you ship to another lab, they can't figure out which lot of virus that lab injected into a mouse, then some of those studies might not be useful for an IND. So it just is a really rigorous process. And I think if you're going to a CRO or industry, that's standard practice in those places. But if it's an academic lab, because we have some more exploratory research work, and then we can move into that translational space. You just really need to have those studies really kind of delineated. And then I think some of the tension too can come to, like, if we've worked on something for 5 or 10 years, it kind of feels like our baby. You know, it's like, oh, we worked on this for so long, and it's hard to give up some of that control. And sometimes it's just, you know, a handoff and you don't see it anymore. And other times it's more of a partnership. And I think if you're maintaining that partnership, for example, if you're working on writing an IND together, which I've done, it's just really kind of clearly defining who's writing what, when, and setting those deadlines and having people's responsibilities really clearly defined is what makes that tension less stressful.
Yeah, Allison, I think you hit on an important part, which is partnership, because we've worked with some groups where we've come in very, very early, and that part is nice, but also when we've come in later. But what's been important is to delineate, this is what you guys are good at, this is what we're good at, let's work together on that. Because I do think it's in human nature to be possessive. I mean, you made something, it's interesting to see it to the end. But again, it's just clear to make sure who's bringing what, because if either one of us had all the answers, we'd just do it ourselves.
Well, and I think, well, maybe Rhonda as well, can you speak to kind of the advocacy group perspective of navigating academics versus kind of industry partners?
You kind of end up the go-between sometimes.
Yeah.
Right. Yes, and what you were commenting on, Allison, is way above my pay grade, so. But I think like early on, we have had some industry partners come to us to like review like what the protocol would be like. Would a patient, you know, actually participate in this? Would they go through this? You know, can they understand it? That sort of thing. And we recently had a situation with a trial where I felt like something that was being asked of the patients was, you know, it was not really practical, and went to the study investigator, and they're like, well, we tried to bring it up, but they really haven't said anything. Why don't you try? So I tried, and they changed it. So, you know, so that's where I think you can kind of work together.
Right. Gets to the, to Matt's point, playing to strengths in the room, which means you have to have an honest conversation about what are the strengths in the room.
Yeah, I think, you know, as we're thinking about partnerships, some partnerships go great and they run very smoothly. Some go south, and you can sometimes feel they're going south or going sideways before they are. I'm curious, you know, without, you know, diving into any too details, but are there some telltale signs that maybe a partnership isn't going well, or maybe we need to shift course before it's too late, or how do you know when you've stayed in something for too long? I'll let anybody jump in.
All right, I'll say something very positive. So, not because there's two Ultragenyx people here or anything, but seriously, with Wilson disease, I feel we are so fortunate that our industry partners have been amazing to work with. They have been so respectful, trying to understand our community, trying to engage with our community. Communication is really well done. And again, hearkening back to my reporting experience, I mean, I didn't see that with like all pharmaceutical companies. So it's been very interesting to be on this side of things, and so we've just had good experience with it. And I have to say that there are two, since I've been in leadership with the Wilson Disease Association, that have fallen by the wayside for different reasons. And I was really sad to you felt like you were breaking up with a good friend, you know, because they had done such a good job with us and were so respectful. But I think that's kind of the message too to industry, is to have it in that way. And you have to also know going into it, you've got to have your eyes wide open. I mean, it's science, and you don't know what direction it's going to go. But if you feel that respect and you feel that engagement, I think it can be a very positive experience.
As a researcher, I always tell my team that people are harder than science. You can force cells to do things. You can give mice an injection. So I always advise people to go with their gut. If you feel like these are good people to work with them. If you feel like it's not working, it is going to hurt your patient population in the end. So really evaluate what you think and take the action you think is necessary.
I think there's two things for me that kind of are those like gut checks, and one of them is I would put them probably in the same category of like trust and transparency. Like you have to be able to have that like trust and transparency, obviously to a degree. Like there might be some things that you can't share because of confidentiality reasons. But really being able to have that open communication. If somebody starts to try to like micromanage the other person, then that relationship can get tense. And so I think having that trust, and when you can, be as transparent as possible. And I think the other thing that helps keep things on track and hopefully reduce the tension is setting realistic expectations and making those milestone driven. So for example, like, in my pathway that I showed in my talk, like, okay, if step one is we need patient-derived in vitro model systems, it's like, okay, so hopefully within X number of months, we'll have PBMCs from this many patients, and then this many months to make the iPSCs, and this many months to do our drug screen, or we're gonna develop a mouse model, we're gonna need this many dollars and this many months. And trying to keep things in realistic expectations for time and cost, and check in on those milestones. And are we on track? Are we meeting those milestones? And if things are not working, that might be good time between those, phases to check in and be like, listen, I'm really struggling with making this mouse model. We've tried a couple and it's just not working. Maybe we need to use another transgenic core, or, just to try to check in on those milestones and make sure everybody is staying on the same page with those expectations.
Yeah, and maybe updating milestones as new information comes along, and goalposts may need to move just because science is science, right?
Science is science, and mice aren't people.
Mice aren't people, neither are monkeys. And I think, Allison, you mentioned this in your talk, the funding climate's changing, right? NIH dollars are changing, there's a lot of shifting at FDA. Industry dollars can be up or down depending on the stock market and cycles. So I'm curious if there's anything that comes up for any of the panelists on kind of navigating those different funding climates and where dollars are coming from and shifting priorities and things.
So I actually, over the last couple of years, have been working with a patient advocacy group. Allison, to your point about how patients feel, within industry, we not only have to show that our drug is working and it's effective, but we also have to show that it has clinical meaningfulness to patients. And in order to do that, that's not always the same scale. So I work in neurology, so typically you would use a neurological scale to show that your drug is effective in the trial, but what does that mean? And in pediatric neurology, for one kid, it may mean that they couldn't brush their teeth independently, and now all of a sudden they can, or they can get dressed in the morning, or they can eat independently when they couldn't before treatment. But how do you get that data? You're not necessarily going to get it from a developmental scale or a neurology scale. And so a lot of patient advocacy groups have been developing patient-reported outcome scales where they've run survey groups. They've gotten those key questions that a mom or a dad or a patient will know that those are the key questions that need to get asked, and they develop them into surveys that you can take throughout a clinical trial. For industry to develop a patient advocacy or a patient-reported outcome scale, it's 2 to 3 years, and you have to validate that scale with the agency, and then you have to show that it's clinically meaningful and it ties in with your data. A patient advocacy group I was working with actually worked with a research hospital to develop this. They partnered with them, ran all the focus groups, got the questions in, helped them with scale development, and then they applied for a grant through the FDA, and they're actually validating the scale. And for our group, that potentially could have so much utility, but also it'll help any company that's working within that disease space. And it's a lot of effort, and it's a lot of work, and it's a lot of money, but you can find grants through the FDA. They're not trivial to fill out. I've done it before. It's a lot of work just to fill out a grant, but it is an avenue that it's a pretty lucrative avenue when it comes to developing patient-reported outcome scales.
That's a good tip.
I would say earlier in the development process, like historically, traditionally, PAGs would provide a little seed funding, grant to get proof of feasibility or proof of concept that then a lab, an academic researcher could leverage for NIH funding. And so we have a gold standard NIH grant called an R01 that's 5 years and usually $2 to $3 million. And then that would sustain that pathway through that whole middle part. But the NIH has just gotten really, really difficult. So, for example, the pay line at NINDS right now is 8%. So that's the Neurologic Disease Institute. And so only 8% of grants are getting funded. So it's just really, really challenging. So if we want to keep things moving forward, we really have to diversify our portfolio more, and that funding from the PAGs might need to just go into later stages, but then it gets challenging because if you fund all the preclinical study, then how are you going to fund the clinical manufacturing, and how are you going to fund those toxicity studies, which are really large ticket items, and that is more difficult to get NIH funding. But the NIH has come up with some new mechanisms. There's this NeuroNext, which is once an IND is submitted, it helps with some of the clinical trial costs. And there's some kind of translational mechanism. So the NIH is trying to develop different mechanisms, but I would say the funding environment is just really challenging. So the role of PAGs, I think, has gotten increasingly more in the stage of development and the amount of money they need to support for drug development.
Just want to remind everybody that there's a mic if anybody has any questions or comments from your own experiences. Thank you.
Thanks for the intro. I have a question a bit about time and cost and what you kind of talked about in your talk, Allison, about picking the right partners. I'm curious if you can list examples of how advocacy groups have saved time and costs. I'm not sure what they can offer to the partners or how they can pick partners, whether it's a smaller lab or smaller industry partner to try to save time, or even if it's sharing, I'm not sure if it's future royalties to try to save like costs today. Any examples that you've seen?
So saving time, I think using some of those like CROs or CDMOs that can do a lot of the actual legwork, that saves time but not cost, because those organizations they tend to be more expensive, but they do it well and they do it right, at least usually, and it takes some of the onerous off the researcher from having to do that. So it could be like, you know, your preclinical study, like dosing all of your mice with your vector, or it could be that toxicity study, but getting that kind of study out of, you know, the researcher's lab, so that usually saves time but costs more money. I would say another place that's like a big decision point is when it comes to your drug manufacturing. So a lot of times a PAG will have to make a decision. For example, you could make like a research grade gene therapy that you're going to try in your mice for your proof of concept, but then you're getting to that toxicity branch, and one big decision is like, do you go ahead and make your clinical product and use that in your toxicity studies, which is in one way the most rigorous way to do it because your actual clinical product is in your toxicity study. But it's more of a gamble, because if you don't pass your toxicity study, you've just manufactured your whole clinical lot. So some people will take a middle road and manufacture what's called like a toxicity batch. And then if you're past your tox study, then you have to go and make your clinical batch. So in the end, that costs more money because you made your research, your tox, and your clinical batch, but it was less risky. So I think there's decisions along the way with who you partner with and how you strategize your manufacturing that depends if you want to save time or money, and those are some very critical decisions to make.
Hello. So I spent quite a bit of my career in small molecule development. This is a little bit newer to me with gene therapy. And in the past, so regulatory agencies or payers, whatnot, quality of life is usually kind of low on the ranking and importance and value that others view your therapy. But here in gene therapy, there's potential for disease changing, and quality of life is an enormous jump versus a marginal one with a chronic disease. So how do we elevate that voice? How do we make those that classically have not really focused so much on quality of life and bring it, right? Your videos were incredibly impactful in that, right? I really felt for the person in a disease that I had not heard of before until this morning. And so looking for advice on that. How do we elevate the patient's voice and the quality of life that they get from these treatments?
Well, what we hear is about the power of storytelling and getting those stories out there. So on our website, we have our videos, we have patient stories, and actually, I feel that we need to do a better job of pushing them out there and getting them on, social media, and so others can see it, and also getting it in front of the doctors. I'll be going to EASL and speaking to a group of doctors, so it's an opportunity to show a story. So I think that's where we have to get creative in getting it out there. The stories are there to be told, and these days you can put video together very easily.
If I can highlight one thing, Rhonda, that I took away from your talk was your baseball card methodology. I thought that was so smart, because once you start to see everything laid out, you really start to see some patterns emerge, and what are those threads we want to pull forward, and maybe the stories that haven't been told enough or haven't been given enough prominence. So I really appreciated that in your talk.
Yes, thank you. And that's kind of that old school part of us coming through. And that was also really good that we had objective people looking at it. I'm so close to my disease. And so having someone come from the outside and really look at that, and when they came, and we had they did half-hour pre-interviews with everybody who wanted to be a video participant. So they came to it, again, fresh eyes. And sometimes you kind of need to do that too.
I was just going to add, from a clinical perspective, there are quality of life scales that are being added to a lot of clinical trials nowadays. The only trouble is there's not a lot of natural history on quality of life. And so sometimes, if you have a longer longitudinal study, then you will hopefully, and if a medicine is working, you'll see a positive trend in your quality of life scale. But they have pediatric versions and adult versions of quality of life scales. So, pushing partners to make sure that this is a critical part and really gets to the clinical meaningfulness side of drug development is really important.
Thank you, everyone. This has been great to have this space together as patient advocacy groups. I'm Amy Christosic. I represent the Med13 Foundation. I was at the Ultragenics Rare Boot Camp recently, and this is kind of like a nice little mini summary of that. My question is, how do we support the PAGs in doing this? We're asking parents to take on this monumental task in addition to their daily caretaking activities and their day job that pays them. And so what infrastructure, what funding, what ways can we help patient advocacy groups do this so that they can be more successful without burning out? I'm sure you've seen, in addition to the success stories that we're talking about here today, some of the failures as well.
The potential for burnout is tremendous. And that's something that we've been very concerned about with having volunteers. And so a strategy that we try to use is, like, for this, we're able to raise funds for this and then get independent contractors to help us out to kind of help with the burden. And they also did so much helping the parents if we're talking about the ELP-FDD, by them interviewing them, they took advantage of AI to help them in writing up their statements, because it's very hard to get them to also do these things as well. And it's difficult for us to kind of spread our base of long-term volunteers for that reason as well. So I don't know that that's answering your question, but it is an ongoing challenge, and you've got to be very aware of it because you don't want to burn people out. And we felt the ELP-FDD brought in more people, for us to kind of tap into to help out.
I know it's really difficult in the early stages, but a lot of the PAGs that I've talked to or work with, they will say eventually, when feasible, bring on at least a part-time, paid employee to avoid that burnout. So Kim Nye from the Test Research Foundation, she could talk to how they eventually brought on someone that can help manage and write the grants and submit the grants. And Mike Graglia from SingGap, they can really talk to where they were getting to that burnout point and ended up bringing on a paid position, and how that really transformed the effectiveness of their advocacy organization, and also helped prevent some of that burnout, because it's real. It's like you're caregiving, you're working, and then you're trying to give out grants and do a natural history study and do a registry, and it's really difficult. So trying to find that support when when the time and funding is there, which I know is also difficult, because you don't want to take money that could be used for research, but if that person is then going to be able to make you that much more efficient that you could apply for more grants, et cetera, then and do more writing or something, whatever your kind of, is kind of your hold up at that time, finding that strength.
I'll just add, also expanding your network as much as you can. Mike Graglia speaks about this quite a bit. You never know who has like an uncle that has $50 million in the bank, or, you know, the patient groups that have done very well often will have an angel supporter here or there. And so I would say broaden your network to get help, but then also broaden it to see if there are other people that can help in ways maybe you wouldn't have predicted.
Thank you.
These were more comments than questions. I wanted to add to broaden your network among the other patient advocacy groups, because learn from what other people are doing. So if somebody's doing an engineering run on a capsid that you're interested in, contact them. There's no point in everybody spending the same money multiple times. So if there are people who are moving into the clinic, how did they get there? Get as much information from them as possible. And from the perspective of saving time and money, my suggestion would be to take a lesson from industry and start organizing grants, not just by aims, but incorporate stage gates and milestones. When does this have to be met? And you don't pay out until it's met. And we're at a time where your dollars mean a lot, much more than they did during COVID. And my other suggestion is to work on something that they call in industry a target product profile. What does your drug need to look at when it goes into a patient? How many cells do you need to hit? What is the amount of correction you need per cell? These are things that companies can build off of and will appreciate if you have that information. And again, reach out to other people who are doing this, because we're all striving towards the same goal, but with different diseases. And people are learning lessons every day that are willing to share. So, thanks.
Yeah, I would say not reinvent the wheel when you don't have to. And one thing that a lot of PAGs are doing now that I love is even sharing their INDs. And so you could get another PAG's IND and not be starting, like staring at a blank page going, how am I going to turn this into 1,200 pages? Like, take another PAG's, use it as a template, use that as your starting point so it doesn't feel so overwhelming.
I would just add, sorry to interrupt you, don't be afraid to reach out to industry partners as well. There are definitely some that are happy to share what they can, either on technology or on some of these more routine things that they have to do. The worst they can do is say no, and then you try somebody else.
Yeah, I was just going to add the Friedreich's Ataxia Foundation. We lean on them a lot. They share everything, and they're a wealth of knowledge. So they're wonderful. So you can learn a real lot from their website.
Hi, everyone. Thanks so much for having this panel to set us off for a good start for patient-focused drug development for the conference ahead. I'm from a rare disease community, X-linked myotubular neuropathy. And Rhonda, thank you so much for explaining and giving an idea of how to host PFDD. I also, for those of you that want to be even thriftier in some ways, we're a much smaller indication and didn't have a lot of money to spend on an organization or a partner to help us facilitate a PFDD, but we were one of the first to have through the COVID era, we were one of the first organizations to host one with the FDA, and we did it all on our own, just using our gifts and skills and facilitation and having families do videos. And actually, one unique thing of doing it virtually was we were able to have many more voices of our pediatric patients, many that use very different technology to communicate. So I just encourage you to even think outside the box even further, to use your resources that you have within the community to do it even at no cost to your organization. And then I would just also challenge everyone to have those challenging conversations. If you have somebody living with a rare disease or a condition that you're working on drug development, I think all of us have had difficult conversations in our lives, and I think sometimes you don't always get to choose the partners you're going to be working with. So really encourage everyone to try to align expectations early and to have difficult conversations when you come up against barriers. There are ways through. Just being brave and having those difficult conversations. And I also really love the conversations around quality of life and quality of life scales. I think this is something that I come across not only in the rare disease work that I do, but also with complex care patients. Quality of life extends beyond physical barriers, and a lot of the current quality of life scales all rely back on physical dimensions, and I think we need to be thinking more broadly of all of the ways families can have their children or their own lives enhanced with these therapeutic, hopeful interventions. So thank you.
Thank you, and I commend you for taking on an ELP-FTD on your own. That's amazing.
So many questions. Well, stick to two. One is, where can I find this uncle of yours? And the second is, could you, the panel say something about partnering with industry and what industry is sort of looking for, particularly when, a lot of rare diseases are, very rare or ultra rare indications and have a very complex biology. It's not, there's still a lot of basic science to be done. It's not clear, and the tools are not together yet, but, without support, it's hard to make advance on that. So.
You want to go?
I can go first, sure. So I would say for patient advocacy groups is try to organize information as much as you can, even if there's not a lot out there. You know not only the disease, but you also know kind of who's working on it. So this, this scientist here in Europe has this idea. This scientist here has this idea. Start to push them to publish that data, even negative data. Get the data out there and start building the foundation for the biology, because scientists look at things and they try to put the pieces together, I would say there. And then also, to an earlier point, around disease progression, start to really, have case studies published that maybe talk about the disease progression. If you can't do that, start documenting or asking people to document. I know of some patient advocacy groups that just reached out via survey and said, about onset or just about disease progression, and start to pull that data together. I mean, I can't speak for all of industry, but I do know that the more data that is available about a disease, the easier of a starting point there is for that. And I also wanted to add one point, which is not directly to your question, but it's just about if industry does reach out, and often industry reaches out to a patient advocacy group and says, hey, can you put me in touch with, X and X expert, or do you know this clinician? Ask for a seat at that table. They might say no, like Matt said earlier, but they might actually pause and say, you are correct, you should have a seat at that table. And I think the more we kind of push that envelope, the more we open the door to having everyone that should be at the table at the table.
And I would say that you were well positioned to help bring people to the table, because you have the most to gain, but you're also the most neutral in many ways, because everyone kind of has either their intellectual stake or their profitable stake, and you just want the result. And so I think that you can serve a really good in-between for all of those things. The other thing I would mention is, even if you are early, don't be afraid to go to industry and ask the simple question of, like, what are you looking for? Because companies might give you very different answers, and one may be more suited to what you're trying to do than not. Again, the worst they do, they say no, and then you probably don't want to work with them anyway.
Well, and I'd say that industry comes to us because they are interested in having a connection with the patients. And so it's almost like we're hopeful, kind of a vetter for that. So there's credibility if we approach somebody, say, we've been working with them and would you like to help? Because we did have a history of kind of like Facebook stalking, of trying to get to patients, and there are some bad actors out there, so it's a way to try to say, no, these are people that you do want to talk to, but also communicating information to our community about clinical trials or surveys or opportunities. So we feel that we're in a position to try to get that information out, and we'd like to think that we're a trusted resource to educate our community. And there are some cases where they've come to me and asked about other researchers. I'll direct them to our centers of excellence, and who they are there, so to kind of open doors for them.
All right, thank you.
Well, I think we are approaching time for our break. We really appreciate everyone's questions and comments, and thank you to the panel for your time and expertise, and to the presenters as well. Thank you very much. We'll plan to resume the second half at 3:15.
No phone.
I know, right?
Sorry, y'all, the lights are quite bright up here. Okay, it is 3:15, so we'll get started with the second half of the programming. So the first half, we're trying to focus a little bit more on that preclinical, non-clinical, the early stage side. Of course, we always want to be thinking about the clinical piece of things throughout the entire life cycle of your programs. But this second half, our intent is to focus a bit more on the clinical side. So when do we actually bring patients, families, individuals with diseases into the clinic and try to learn as much as we can from them? So we have two amazing speakers lined up for you, and then a similar setup where we'll have a panel discussion at the end. So our first speaker will be Dr. Scott Demarest. He's an assistant professor in the Department of Pediatrics at Children's Hospital, Colorado. And he's going to be doing a review of clinical research pathways, the NIH investigator-initiated and industry-sponsored trials. Followed after Scott's talk will be Ms. Wumi Bakare, and she's the founder of Sickle Cell Prodigy. She's going to be going through a pivotal decision points for participants with sickle cell disease. All right, Dr. Demarest, you can come up. Thanks.
Well, thank you for the invitation to speak. If I'm not making eye contact, it's because, like, we can't look above about right here without being blinded. I'm going to try to give a little bit of a framework about how I think about aspects of getting from preclinical to clinical and that translational piece, and then we'll have plenty of time to discuss specific details in the panel session later. So I've broken this up into three main parts: clinical trial readiness and what we can be thinking about even early, pathways to get from preclinical to clinical trial, and then some of the strategy components that we need to think through. And I'm really speaking from assuming the audience is patient advocacy groups, so this is a PAG perspective on how to approach this as opposed to an industry or academic perspective. List of disclosures for various groups that I've worked with, and most of this content is actually based on my own flawed perceptions of the world. So this is just a little bit of a context slide, which is to say, like, this is happening. So this is out of a report put together by the Children's Hospital Association, which outlined just a couple of months ago that there are 264 cell and gene therapies that were actively in the clinical phase. I live this every day. This is my entire job, and that was surprising to me. So this is happening. The anticipation, despite all the bumps, all the challenges in investment and everything else, is that there's a 10xing of this market in the next 10 years. So it's the right time to be thinking about all of these things. So just thinking about clinical trial readiness. I'm going to go through a couple of the components of what clinical trial readiness might mean. We can dig into these discussions in more detail, but what are the components one will consider when thinking about clinical trial readiness? And I want to frame all of this by starting with clinical trial readiness is not a binary phenomenon. You are not either ready or not ready for clinical trials. You can go through a variety of steps becoming increasingly ready for a better, well-designed, more rigorous, more likely to be successful if the product works, clinical trial, but it doesn't mean you can't run a successful clinical trial if you haven't done all of these components. So the first one is basic biology, and this kind of is coming back to the preclinical components to a certain extent, but you have to understand the basic pathomechanism of the disease. You have to understand whether this is a gain of function, loss of function scenario, a dominant negative. Dr. Bradbury alluded to some of these components right at the beginning of her talk as well. This really is where everything starts, because you don't want to replace something if this is a gain of function, as an example. So your whole strategy of developing a therapeutic has to be predicated on the right understanding of what is fundamentally going on. Are all the patients in the population the same mechanism? There are numerous genetic disorders that have multiple mechanisms of disease, so this is a critical question. And I don't mean to say you have to understand absolutely every facet of the biology and what is happening as a result of a dysfunctional gene, but you do have to understand some of these basic principles of the neurogenetics. Thinking about patient identification, again, this was alluded to, so I don't want to talk about this too much, but there's kind of different levels here as well. So you could just say, we know where the patients are, we know who they are, we can reach out to them. Incredibly valuable. Also sort of the bare minimum of what it means to be able to start to think about a clinical trial. The next stage would be able to think about sort of more rigorous natural history, and that can achieve several goals. So you might be thinking about a natural history for understanding what outcome measures are going to be appropriate for that population. I'm going to talk a little more about that. Understanding how the presentation changes across the lifespan or as it progresses. And then the ultimate, particularly in rare diseases and more importantly in ultra-rare diseases, is to think about having a data set that could actually be used as an external control cohort. That's the most rigorous, the most expensive thing to try to get to, right? And so, again, there's not one answer. There's a spectrum of how ready you are for a clinical trial. And some of these pieces are incredibly valuable, like basic understandings of how the presentation changes through the life course. You can't really design a good clinical trial with good exclusion and inclusion criteria and endpoints if you don't understand those basic components. But if you have actually been able to prospectively map it, you're in an even better place and maybe in a place where you could use an external control. So when we think about endpoints, what are we trying to accomplish with our endpoints? Well, the first thing is they have to be relevant to patients and families. The FDA anchors to how patients feel, function, and survive. That's what we're always trying to assess with an outcome measure. And patients and families really have to be the guide about what is meaningful, what is capturing things that actually matter in their lives. It's great to be able to say we have a validated endpoint, but if it's not relevant and doesn't capture something meaningful to the patients, then it's not a particularly useful endpoint from the standpoint of the FDA's perspective. And so that's where your voice is absolutely critical. Not all things that are important to patients are a good endpoint. There's some things that are really important in terms of clinical care that just don't scientifically make good endpoints for a clinical trial. An example of that could be something like scoliosis. Scoliosis has a big impact on life. It also is something that tends to happen around certain ages. You have to wait a long time to see whether you prevent it. Like, there's challenges to thinking about that as an outcome measure, but it's a clinically important feature. There's also the reverse. There are some things that are good endpoints which may not be meaningful to patients and families. So we have to think about it from both perspectives. So outcome measure validation is something that's discussed a lot, and this is similar to clinical trial readiness. There are degrees of validation that one can go through. The FDA has provided guidance on validation of outcome measures. I will say, and they even have processes that you can put measures through. Those are really not designed for rare diseases. The bar is actually kind of unreasonably high based on conversations with the FDA. They'll say that themselves. You just need way more patients than tend to be available in rare diseases. But there's a couple of things to kind of think through. So are there existing measures? It's a lot easier and cheaper to repurpose or adapt existing measures to a population than to create new ones. I say that as someone who's made a significant portion of his career making new ones. You need to do that in some cases, but it is more time and more effort to do it. So if there is something you can repurpose, that's great. Even if you are repurposing or using something that has been validated, meaning you know the psychometric properties in a different population in almost every case, right? It's not been tested in your population, it's been tested in something that may be similar or less similar to your population. It may have been used in trials as an endpoint previously, so the FDA may have familiarity with it. Those are all things that are useful, but one still has to consider what does it look like in the population that you're planning on utilizing it. There can be curveballs sometimes that certain items or ways that you're approaching it don't work well in all populations. So how deep you go on that validation within that population that you're trying to use it for is a little bit of a strategic discussion, but sometimes it's as simple as we're going to do it in a cross-sectional way. We're going to get as many patients as we can. We're going to do it one time, and we're going to look for things like, is there a clear ceiling or floor effect, which means you're got a lot of people at the edges of the score, not in the middle of the score. That could be problematic. All the way through to redoing psychometric analyses and statistical analyses to say whether or not it fits well in that population, and to even doing it then prospectively over time to look at how stable it is across different time points, or whether it's really noisy and bounces around a lot within that particular population. These are all increasing levels of validation you can bring, but you don't have to necessarily do all of those things in order to jump into a trial. The more you've done, the more informed your trial is going to be and have useful information. So clinical trial readiness comes down to kind of a few components, and this is a bit of a summary slide that I'll kind of bring through. So validated outcome measures, validated biomarkers, where possible. Biomarkers are not mandatory, but are awfully nice to have. It makes things a lot simpler when they're good. When they're spurious biomarkers, meaning they're not really related to the fundamental biology or to the outcomes of interest, you can get into problems with biomarkers. But if you have them and they're good, they're really useful. Understanding the longitudinal trajectory and phenotype of patients, the range of them over time, and then having a network that is able to actually run a clinical trial. Some people made a reference to things like centers of excellence. When you have centers of excellence, you have people who are familiar with the disease, hopefully familiar with the types of outcome measures you're going to use. They're kind of ready to go to jump into a clinical trial, as opposed to trying to figure out who are going to be the sites and where are we going to run this. I alluded to, obviously, the spectrum of trial readiness. And so on one end, you have a basic understanding of the phenotype across the lifespan. I would argue it's very difficult to do any kind of clinical trial design without understanding that. Can you measure some of these features reliably? So you've assessed some of your outcome measures and you think that they're going to work, not just in general, but in this population specifically. Are you able to perform a symptomatic trial? So we're at Cell and Gene Therapy Conference, so we're talking about disease-modifying treatments. But in some cases, you may run an epilepsy trial, for example, or some other sort of symptomatic trial. And that still teaches you a lot about the disease and the ability to run a trial, even if that has different endpoints than what you would plan to use in a disease-modifying trial. If you had a successful symptomatic trial, that's even better in terms of thinking about readiness. Validated measures across all key features, so it's great to have an outcome measure that catches something. Most rare diseases and genetic disorders have more than one domain of impact, and so are you capturing lots of different things in a comprehensive way, plus minus biomarkers, and then sort of the pinnacle would be that you actually have a longitudinal trajectory, high quality external control cohort. This is only achieved by having a prospective, highly rigorous natural history study. And that is, of course, the most expensive thing you can do within trial readiness to kind of get to that point. So how can you pay for all this? So there's a few different routes. I come from a neurology background, so I have a little bit of a neurology-centric view of the world. But there's certainly things like the Rare Disease Research Network. There's UO1s from the NIH for clinical trial readiness. There's FDA grants associated with this. So there are, and these are still being awarded, government-funded mechanisms for this. Obviously, you all, there are sources of funding, and there are numerous examples of advocacy groups, some of which I know are sitting here in the audience, who are funding this type of work. And funding some of this doesn't mean you have to necessarily fund all of it. So there's an opportunity for you to be strategic with your resources around how can you either initiate with the plan that maybe you're more attractive as a grant recipient for your community in the future. I worked a whole lot with CDKL5. We had that exact mechanism. We had 1 year funded to start our work, and then we were able to successfully get a UO1 rare disease clinical trial readiness grant that allowed us to have 5 years of funding and really run the prospective and all of that. Being able, there's a like catch-22 in the grant world for those of you who don't live in that all the time, which is like you kind of have to know the answers you're going to get before you actually do the work that you're trying to get paid to do. So there's a circularity to all of this that your little bit of seed funding can help your group be a lot more attractive for a grant because you're further along in that process, which makes it seem like paying for you to do that work is less risky, even though you haven't done all the work yet. And so a little bit of seed funding can go a long way when it comes to NIH funding. You can also think about sort of, can we pick off specific critical gaps that are relevant for our community that you want to try to attack? So maybe endpoints are the biggest problem. I don't feel like any of the endpoints represent myself or my patient or my family. You might really feel like biomarkers are the critical component for your group. It may be that you want to invest into centers of excellence to sort of set up that network concept. You may be able to fund a cross-sectional or retrospective study that's trying to get some of that basic description of the disease. That's a fraction of the cost of what it would be to do the prospective version, right? So you don't have to do the whole thing, but assessing where are you as a community and what are some of the most critical components can be helpful. Certainly industry also is a funder of these types of studies. Typically that is when a company is already relatively committed. So they don't usually want to spend that money until they know that they have an asset that's going to go towards trial, and they're pretty confident about that. They also tend to want to own that data. You can fight with them on it. You may or may not be successful, but you can try. But they tend to want to own that if they're paying for it. So then we think about sort of launching a clinical trial. What are the things that are necessary? I don't need to talk about the top. That was the whole first session, all the things you have to do in the preclinical realm that have to line up. But obviously those things have to inform the way that you're thinking about your clinical trial, including things like dosing, levels, et cetera, et cetera. I think it's useful to think about who's on the team. So I've kind of put some thoughts here. So obviously you guys, so patients and families have to be on the team and have to be really a loud voice in that, providing input. And we can talk about sort of which ways and models would that look like in terms of your role. You need some disease experts that can provide relevant context for all of those components we talked about. When are certain problems more of an issue across the lifespan? You guys also inform on that. Are there any other biological things we need to worry about when we're thinking about treating? You want some clinical trial experts around the design, timelines, cohorts, inclusion-exclusion criteria, endpoints, regulatory experts that will guide what is today and tomorrow's thinking at the FDA. Next week it'll be different, but the best we can do to try to think through how is the FDA going to view the way we're proposing this. There is an element of like you put it in front of them and you find out and they will tell you what they think about it, but having some expertise in that realm can be super helpful. And then obviously you want to have some idea of sites that would be capable of running these types of trials. There's lots of critical partners that you want to think through: funding, CROs, sites, analytics. CROs are an umbrella term. Dr. Bradbury referred to CROs in a very different way than I am. CROs also do implementation of rigorous clinical trials as well as preclinical CRO work. So there's kind of different types of CROs that may function in those spaces. But again, tend to be more expensive, but also more consistent about rigor. And when you're thinking about data that will be acceptable to the FDA, rigor is really important. So back again, how do you fund the clinical trial? So there are all the same options that we talked about on the natural history or clinical trial readiness components that you can think through. So government, patient advocacy or other nonprofits, academic, and industry, and I'll kind of take these each in turn. So again, there are various grants that may be relevant for a particular clinical trial from NIH, RDCRNs, often have either little pilots or can have some sort of clinical trial component to them. ARPA-H has gotten into this space with the launch of their rare disease efforts in recent times. Those tend to be a little bit more like high-risk, high-reward types of development programs. And then there may be various clinical trial networks that are funded by the government. NeuroNext is an example. Children's Oncology Group has various funding that can support some of these types of trials. Within the patient advocacy group and nonprofit space, it is possible to fund a whole trial. So particularly as we're thinking about sort of rare to ultra-rare disease trials where the number of patients may not be a huge number, it is possible, and there are examples of advocacy groups that have really taken that on entirely on their own. There may also be a version where you're thinking about just kind of how do we get it started? So can we de-risk this program to make it more attractive to industry partners by getting through the phase 1, or thinking about an N of 1 or an N of few sort of trial, and we could get into plausible mechanism and all those types of things if it's helpful in the discussion, but that's really just sort of an ultra-rare application of existing statute within the FDA and thinking about that mechanism. And it's just a note there that huge trials may not always be required, and I think that there is a shift to starting to think through what does it look like to do highly rigorous, convincing, safe trials with a smaller and smaller number of patients. But there's a lot we're still trying to push the envelope on and see how far we can take that as a community. Academic partners may be interested in funding portions of trials. They are usually a required partner in sort of all of this, even if it's industry funded or it's funded by a patient advocacy group or the NIH. Academia is usually involved, in some form or fashion. If they actually have IP involved in some form or fashion, that tends to make them more interested in also putting up their own funding for certain aspects. And you can always work on sort of investigator-initiated trials. That may be a way that you can reduce expenses in certain cases for a trial, whether that's for proof of concept or otherwise. You do have to be a little bit careful about data quality on investigator-initiated trials. In the rare disease space, every patient treated matters, and matters a lot. And some investigator-initiated trials are much more just compassionate use, and the rigor is not there to make that data as useful for a regulator eventually. So you again, clarity of what you're trying to accomplish. And I just put here champion, like it's really valuable to have a champion in the academic space that's willing to kind of go to bat for your community, try to help to make that happen. It's necessary across any of these types of models. Industry involvement. I did this with a lens of like, the patient advocacy group is still driving this, but realizing there may be different levels of driving. What is the relationship between the patient advocacy group and an industry partner? So there's one where you kind of are saying, I have the most control over this. You start your own startup, so you become industry as part of a patient advocacy group. And again, there are numerous examples of that having been done. You retain IP or licensing associated with it, but you partner with someone to actually bring that forward to clinical implementation. Or you're really taking the attitude of, maybe we're not going for just one product. We want multiple products or different shots on goal, and we want to de-risk the entire space for our disease and have various partners who will bring that forward. Each of these have different strengths and weaknesses associated with them in terms of how much you're putting out from a patient advocacy group perspective, how much control you have over the scenario, whether or not something can get put on a shelf or not, but also the amount that you are putting your own money behind what you're trying to accomplish. So there's a give and take there. So how does that all kind of come together to think through strategic decisions? And this is not, there's no judgment associated with any of this. I'm kind of putting the whole table mat out there of options. And the point is for you to think through what makes sense for your community. So most important thing is, what's your goal? What are you trying to accomplish as the leader of your patient advocacy group? What is most important to your community? Your goal might be, I want to treat my kid or myself. I'm a pediatrician, so I tend to think about patients as kids, but I recognize that there are adults who are also in this position, so I don't mean to ignore that perspective. If the goal is really, I want a treatment in my kid as quickly as possible, compassionate use may be reasonable. That doesn't necessarily have some of the long-term sustainability associated with it, impact across a broader community, but it may be what matters to you. Are you looking for a single treatment for your disorder? I just want to get one treatment, one option for my community. Or are you thinking about what's the 20-year view? Multiple treatments, iteratively improved over time that are really shifting the bar. I get that for some, that is like, I can't even imagine a year from now, let alone think on a 20-year view. Cystic fibrosis is probably one of the best examples of a group that has managed to have a sustained impact with iterative improvement in terms of treatment options for their community. It can really go a long way, but sometimes it's hard to be thinking that far in the future when you have what's in front of you today and you're trying to do the best you can to treat. So there's a balancing act there. Critical questions in that: how much time do you have? How far out are you looking? How much time does your community feel like it has? This makes a difference in terms of whether you're talking about static chronic disorders or degenerative disorders and things like that. Impacts a little bit how you approach some of these questions in deciding what makes sense for your community. Rigor is incredibly valuable. Having rigor behind the data, and when I say rigor, I mean not just that you know the data, you know the source of the data, you know whether data has been changed at any point. These are all like criteria set out by the FDA when it comes to clinical data for them accepting, either for clinical trial readiness, external controls, or for clinical trials accepting that your data really is what it is. There's things like Part 11 compliance and stuff like that that have to do with the way you structure databases, collect data, monitor that data to know that it is reliable. That's really, really valuable if you're going for a label-enabling type of scenario. You're trying to get something across that finish line to commercialization. It is expensive. It's more time-consuming to set up. It takes resources. So you have to kind of think through how you can manage that. You have to be realistic. What do you have in terms of resources? What can I do now? What can I aim for in the future? How can one thing maybe snowball a little bit? Can we get this step further along, and then that opens more doors or de-risk or brings in more interest for investment to kind of keep things moving? And then this goes back to that original question I kind of said. So how much control do you want to have over the process or the product? And for some groups, that is like really clear cut. Our group is a product, and we are bringing that product all the way through, and we want control over it. For others, they are on the other side of that spectrum and say, we want to have multiple products coming through. We don't have to have control over all of them. We want to de-risk this entire space for our community so that multiple shots on goal may come forward. Both have advantages, and it's really deciding what makes sense for you and for your community. So this probably isn't new to this group, but just kind of like basics of the clinical trial pathway and how we think about this. So we spent the first half of the session talking about the left two, which is sort of preclinical development of a particular product. And this is the traditional clinical trial pathway with phase 0 all the way through phase 4. Getting through phase 3 is really the critical component, which allows you to get to commercialization, and then phase 4 is post-marketing follow-up and understanding what it looks like in the real world, and sometimes long-term safety and efficacy. Increasingly, within the rare disease world, these to get smushed together a bit, and there's good precedence for this with the FDA and guidance from them about how we approach rare diseases that allow you to think about, rather than phase 0, 1, 2, a phase 1, 2 smushed together where you can find the right dose relatively efficiently and begin certainly having a sense of safety, but also even a little bit around efficacy. And then increasingly, we're even starting to think about, can that phase 3 even be bundled into that? And part of the kind of ultra rare disease scenario is that you're kind of smushing all of this together, and you really are then starting to think about, how do we prove to the FDA within a small number of patients, small, like 10 to 20 maybe, that we are convincingly both establishing safety and efficacy. The FDA is not allowed by law to approve something that hasn't established safety and efficacy. That's their mandate. And so you have to make that argument to them. And in order to do that, generally speaking, when we're talking about cell and gene therapies, placebo-controlled is not usually on the table. So you have to have a really rigorous external control that has all of those data qualities and rigor that I was talking about that allows you to look at that. Some cases that can be an extended lead-in study where you are actually enrolling the patients that are going to be treated into a natural history and then drawing from that. And then patients can act as their own control as well as to a small control cohort. So there's ways that we can get more rigorous about some of these things, but I think it's really important when we're thinking about these and plausible mechanisms kind of outline some of these concepts, but really they are just talking about how do we apply existing statute to ultra-rare disease. The rarer the disorder, the more compressed this tends to become, and planning becomes that much more critical. Oftentimes in a traditional method, we find what we call signals in the phase 1, phase 2. So we focus the phase 1, 2 on safety. And we go, great, like safety was okay. And while we're doing that, we're like, wow, what outcome measures showed efficacy? Like we have some ideas, but like, okay, what really showed up and actually gave us a good signal? And then I can choose to put my chips down on that one as the best endpoint in the phase 3. And when you're shrinking this, you kind of don't have that luxury. It doesn't mean you necessarily have to pick what it is, because I think the FDA, this is all my own flawed opinions, I think the FDA is going to be reasonable about assessing what you have in terms of data, the totality of evidence, they like to refer to things, but you can't just make it up later, right? You have to know, like, these are the things that are important, and I'm going to collect them highly rigorously, and then we're going to show where that signal is in as few patients as possible. And that means every patient has to provide valuable data, and that means you have a really clear plan about it going in. So I think we kind of up the importance of planning and making sure we're really clear about what we're trying to accomplish, the more we try to shrink these traditional phases into single phase sorts of protocols, even if they have sort of different cohorts that allow you some flexibility to shift across those. You're trying to do it kind of all within more or less one study with maybe a couple of different cohorts in it. So we can chat more about all of those things, but just kind of sum up the way I think about this. First of all, know your goals. There's no strategy without a clarity of destination. You're just sort of randomly doing things otherwise. Who are your partners? Alignment and trust are critical with your partners. Agreements, meaning legal agreements. This is the trust but verify. So I think that trust is really important. I don't think you can have a good partnership without an aspect of trust. The legal agreements are the verify part, and they're important for making sure that when I say this, you mean, and we're thinking the same thing when we both say these things, because sometimes that's not the case, and you want to find that out early, so trust but verify. Are the partners that you're bringing in the right partners for the task at hand, what you're asking for them to do, or is it a different partner that needs to cover some component of that? And that takes some critical assessment. And we should be really honest, this was alluded to earlier, every partner has their own goals. They may also have shared goals. I think everybody wants patients to be treated and it to be effective. Like I think we actually do legitimately across the board. I don't know any evil people that I've met in this that are like, I want people to get worse, right? We want patients to get better, but they have other goals in addition to that. And so you want to be honest about what those goals are. I have them myself, even though that is clearly my most important goal is I want to help patients get access to treatments that can be life-changing for them. But I also am an academic person. Like my job gets judged by publications and by grants, and like I have bosses that I have to, respond to and get to tell me what to do. And so I try to put those things second to patients, but I still have other things that are going on, and that's true for everybody in the game. And then sort of thinking through the regulatory and the legal, Aspects of FDA engagement and approval, people have said several times, early and often, so engage with the FDA, both for sort of PFDD, which I consider in some ways to be part of that clinical trial readiness component, so thinking about and making them aware of your disorder and community, but also your pre-IND packages, et cetera, that you can get feedback around. Aspects of regulatory things like IRBs, clinical trial agreements, these things are really slow, they're painful, they're gonna be frustrating, they have to happen, and you have to work through them, and you have to be realistic and plan for them. And then I think there's actually really important questions for you as groups to think about of what your entity looks like. What should your legal structure be like? Does it make sense to be a patient advocacy group or some other kind of nonprofit? There's public benefit corps, there's for-profit, there's transitioning between those at different phases. We do that on the academic side as well. We develop something, spin off companies, people either go run them or they license it or do whatever else. It's okay, but the legal entity as a driver of something has to fit to the purpose that you're trying to accomplish. And so it's worth thinking about that. And I say questions, but that's a lie because we're going to do that at the end. But I can't come up here without shamelessly plugging our Precision Medicine Institute at Children's Hospital Colorado, where we have the Center for Innovative Genomics and the Center for Transformative Therapeutics. And we think this is really the virtuous cycle that's going to allow us to move the bar and help support patients and families within the pediatric space getting to these types of treatments. Thank you.
All right. Thank you so much, Scott. Next up, we've got Wumi.
Hi, everyone. Before I start with introducing myself, I wanted to say thank you to the American Society of Gene and Cell Therapy, as well as the National Organization of Rare Disorders, for having me today. My name is Wumi Bakare. Doesn't look the way it's spelled, but to woo a lady, woo me. Hopefully you won't forget. I was born with sickle cell disease, and then 6 years ago, my life transformed. I received a stem cell transplant at the National Institutes of Health. And so I stand before you today as a lived experience expert. I'm not a researcher, not a clinician, but I know this walk far more than some scientists do. Couple of disclosures. There we go. I am a compensated member of the Patient Advisory Board for Beam Therapeutics. They are a company that has a base editing therapy for sickle cell disease. I wanted to be very transparent about that. The views I'm expressing today are my own. They're from my lived experience, but also the work I've done. Sorry, I just realized this is really low and I'm quite tall. But also the work I've done through my advocacy organization, Sickle Cell Prodigy. Let's start at the very beginning. I think it's a very good place to start. All right, the base of my presentation today is going to be founded, the bedrock is my life story. But I guarantee you, by the time I finish the presentation, it'll all start to make a little bit more sense. So I was born in Lagos, Nigeria. I'm not American. And growing up, I had to navigate stigma and silence. Even though my home country has one of the highest sickle cell populations in the world, it's a highly stigmatized, demonized, and weaponized disease. The healthcare system doesn't center sickle cell disease, and so much of my life, I navigated sickle cell on my own, but with the support of my ever-loving family. Couple of things I want to share is that in 2015, what started as a vacation to Seattle turned into a nightmare and a little tussle with my own mortality. I was diagnosed with something called Mycobacterium avium complex. It started in my liver, spread to my kidneys, my lungs, and my entire bloodstream. At the time, I was on the standard of care treatment for sickle cell disease, hydroxyurea. Was not benefiting from it. And so as a patient, I did my own clinical research and started looking into transformative therapies for sickle cell. Now, the story sounds like smooth sailing, but it really wasn't. In essence, I contacted the NIH and started to express interest in cell and gene-based therapies. And the options were provided to me. I could either go through a haploidentical transplant where one of my parents would be my donors. I could go the route of my brother and go through an allogeneic stem cell transplant or gene therapy. Realistically, for me, gene therapy was far too new. I didn't want to take that risk. And so my older brother, who's captured there, was my donor. Now, I will share this. In my decision-making process, something happened. A really good friend of mine had gone through a stem cell transplant and passed away. That halted my decision-making. Because even though I did my risk evaluation and I knew the science, I understood it, I was at a critical point where I started to think, if she passed, what would make my story different? And this is the reality that many patients face in their decision-making process. But what I will say is the big important aspects of these conversations with clinicians were the knowledge gaps that existed. No one told me navigating my mental health on the other side of sickle cell disease would be one of the hardest tasks I've ever had to do. And this is the analogy I use. If you are married for 30 years and you become Mrs. Black and your husband passes, if someone sees you on the road and calls you by your maiden name, you're less likely to respond because for 30 years, you've been Mrs. Black. It's the same with sickle cell. I was born with sickle cell. It's all I've ever known. So now, navigating life on the other side of this disease is a path less traveled. Oftentimes, I am scared and it feels dark and lonely. I share all this today because it's going to highlight some of the decision-making components that many patients and their care partners are dealing with. All right, so what was beneath my decision? In all sincerity, I had reached the age 30. I had been navigating sickle cell disease for 3 decades and nothing seemed to be working. But my top concerns were death. Everybody would say that's a valid concern, right? Right. The second was fertility. Conditioning and radiation, the chemotherapy and radiation process impacts fertility. At the time, I was unmarried, didn't have kids, still don't. But I always wanted to be a mother. And so I had to have the hard conversation with my parents of, I would like to freeze my eggs before I embark on this journey. Let's just say they weren't in support, but we got it done anyway. The other case was trusting in the system. Sickle cell disease primarily impacts communities of color, people that look like me, and health systems weren't designed for people that look like me. And so over the years, I had experienced a lot of mistrust and implicit bias in emergency rooms where, as a patient, I should feel safe, but didn't. And then there was the ever-triggering question of talking to clinicians, they explain the science, it sounds all cool, like X-Men. And then you ask the question, what is my life going to look like on the other side? And the clinician is like, due to HIPAA regulations, I can't connect you with the patient who's done the treatment. But as a patient, I wanted to talk to someone who had walked the walk and talked the talk. The science is great, but from a lived experience perspective, I needed to know what exactly I was going to face. Now, realistically, for sickle cell disease, I'm going to be completely transparent. Our decision-making is not cell and gene therapy versus nothing. We have 3 disease-modifying therapies currently. Hydroxyurea, which is the standard of care treatment, crizanlizumab, which is an infusion, and then chronic transfusions, red cell exchanges. They bleed me out of one arm, they transfuse me through another arm. But then the CGT modalities, which are ever present in conversations these days, I want to guess that everybody in this room has seen the first patient to get cured of sickle cell in New York. You probably all saw that story. It made its rounds. And so there's Casgevy on the market and Lyfgenia, but their price tags are $3.1 versus $2.2 million. And of course, the route I took, which is an allogeneic transplant. But what patients are critically thinking about is the side effects. We all see movies and media depict chemo as you lose your hair and there's a little bit of hyperpigmentation, but what no one prepared me for was the short-term memory loss. Sometimes I'm in a kitchen, I don't remember why I'm there, and sometimes I'm on a call and I can't spell kitchen. But no one told me to expect that as part of the conditioning treatment process. Now, let's talk about my life now. I'm 6 years post. A celebration. Thank you very much. But the reality is one of the hallmarks of sickle cell disease is chronic pain. I will be completely transparent. Selfishly, I wanted to get rid of pain. It had taken center stage in my life. But the reality was, even though there was a change in the hemoglobin pathology, my nervous system re-anchored pain. And so now I still live with chronic pain. It's just the only difference is it's not classified as a vaso-occlusive pain episode. So now I'm having to define and characterize the type of pain I feel. Let's also talk about the disease-related complications and everything from avascular necrosis, there's bone issues, there's organ issues I've dealt with over the course of my life. Those don't end because I have experienced a functional cure. I'm doing that in bunny quotes because one of the things you'll notice in my presentation today is inherent in the word cure is finality. Cell and gene therapy is not a finite bibbity-bobbity-boop moment. It does improve the patient's quality of life, but with a chronic illness like sickle cell disease that's progressively debilitating, as you age, your disease becomes slightly more complex. And of course, this varies from patient to patient because there are 5 genotypes of sickle cell disease. I just happen to have the most severe type, hemoglobin SS. Now, survivorship is a new clinical state. Currently, there is no standardized framework, and that is where I come in. That has been my life mission over the last 5 years. There are many survivors like me who are grateful they had these treatments, but who are navigating a path that clinicians and researchers didn't necessarily build for us to exist in. I will say this and say this loud and clear. Cure is not the end of any patient's story. It's just the beginning of a new chapter. We are really solving the wrong problem. And I think when we think about cell and gene therapy decision-making, it's not really about like choosing treatment as it is navigating that treatment continuum of before, during, and after. All right, so for the science folks in the room, here's the deep dive into a literature review. So 3 critical things were highlighted when I was preparing for this presentation. Number 1 is decision-making. Not all sickle cell patients want this transformative therapy. Reasons being because disease burden varies from individual to individual. So most patients that are exploring cell and gene therapy are having a limited quality of life. And the disease burden has been not just impactful for them, but also their family and their entire community. So that is one of the primary reasons why patients explore cell and gene-based therapy. Now let's talk informed consent. I have a science background, which is why I can be excited about the science, but the average sickle cell patient does not. They don't work in healthcare. They understand the healthcare system from a patient perspective. And with sickle cell disease, many patients experience silent strokes. So in the process of informed consent, patients don't really know what they are signing. They're just so desperate for a cure. And last but not least, access and equity and financial toxicity, the hefty price tags on these new gene-based therapies. And the reality is 50 to 60% of US patients with sickle cell disease are on Medicaid. And even though there is a CGT CMS model, as of 2025, less than 110 patients have initiated treatment. Let's stop and think through that. Less than 110. And this disease impacts over 100,000 patients in the US. That speaks volumes of the hesitation that is coming from the patient and caregiver community. Now, I wanted to pause on these two quotes because they are deeply personal to me, but they also reflect what survivors in this community are highlighting. I wanted to talk to a patient who has been through treatment, but no one was recommended to me. Another patient, I was desperate for a cure. I'm on the desperate for a cure list. I saw this as an end-all be-all. And I think in the critical education of patients, we often need to help them set realistic expectation. The burden should never be on a patient to figure out what their life is going to look like. I think we need to have more open and honest conversations with both patients and their support community. Now, realistically, we would like the decision-making process to be linear, right? We hop from this to this to this to this, and we're done. But the reality is, one of the things you'll notice on these next two slides is the arrow goes in different directions, because at any point in time, you can think you're heading to the finish line and something can pull you back. Now, most patients start with an awareness. You see that news story. In my case, my aunt sends me a WhatsApp message of the first patient to be cured in Louisiana. And again, I have the awareness that these cell and gene therapy treatments exist. Then information seeking, and for many patients, this starts with community. It often doesn't start with clinicians because, like I said, this specific patient population, we don't necessarily trust healthcare systems. And so this will start, the information seeking process usually starts with town halls or webinars that industry does to better educate us about their products. Then you do the risk evaluation, where you critically start thinking about, if I do this, what do I do if this other thing happens? I have patients who I've spoken with, pediatric patients who have gone through these treatments and now have an autoimmune disease. So you really critically need to think of, if this has alternate effects of my body, can I live with the decision that I made? And then social consultation. I will be completely transparent. For the Black and brown community, our foundation is our faith. And so this is where the priest and the pastors enter the chat, as well as your loved ones, of critically helping you decide, is this treatment right for you? Now, using my story as a case, at this point, my father's side of the family, who are Jehovah's Witnesses, were frowning upon my decision to embark on stem cell transplant. Because for them, they looked at it as getting a breast augmentation. They were like, this is ridiculous. If God wanted you to be with sickle cell, live with it for the rest of your life. And so for some patients, these are critical voices that they have to consider in their decision-making process. And then, of course, you get to the decision. Do you proceed? Do you defer? Or are you in a position where you're like, this is great, but it's not for me? Now, in moving to the next slide, you see that there are loop-back triggers, right? Where I mentioned before that at any point in this decision-making, something can make a patient pause. In the story I shared earlier, I mentioned my really good friend who passed away after transplant. And that was a halt in my decision-making process. But for other patients, it's coverage of my insurance told me they would be able to cover this cost, and now they've changed their mind. And that could halt the decision-making process as well. There's also confusing or conflicting stories where maybe there are new clinical data that's been released, and then the patient is like, whoa, 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 whoa. Something new has now come in the news cycle, and now I need to be a bit more cautious so that I make a decision that is not just mentally sound for me, but something that I can live when an influx of other patients and loved ones are asking me questions that I need to be able to answer post-treatment. Now let's talk about where the system fails. Patients are really in a position right now where with the era of social media, there's a lot of quacks online. People who pretend they are the know-it-all on all things cell and gene therapy, who could mislead you down a very challenging path if you're not careful of how you decipher information. But we also need to critically think about, do these patients understand the difference between gene addition, gene editing, or are they lumping these new treatment options into the umbrella of gene therapy, not realizing that during the process of harvesting stem cells, you're the donor. It's not like a stem cell transplant where you have an external donor. You are battling disease, and so during the stem cell harvesting process, there might be a chance you don't even have enough stem cells that can be modified using gene editing. Then we also talk about clinical versus perceived eligibility, which is critically thinking about they tell you if you go through IVF and freeze your eggs, you should be good when you get to the other side of this treatment. But I know 2 survivors who are close personal friends who have gone into early ovarian failure in their 30s. That wasn't clearly discussed. And now, even with them freezing their eggs, they will never be able to biologically have children on their own. Then we need to consider trust and survivorship. If patients are in positions where they cannot see other survivors thriving, that impacts how they critically think about cell and gene-based therapies. Now, let's not even add the added component of dealing with pediatric patients, because a lot of the examples I've shared with you today are from adult survivors. But when we think about the critical patient, pediatric patient component, we think about the process of assent, which is you're making a decision as a parent on behalf of your child. If your child is 10 and hasn't started their cycle, are you in a position to make a decision where you don't preserve fertility? And if the child on the other side of recovery cannot have children, you as a parent have to face that child and say, well, I wanted you to get a functional cure that I took your ability to have children in the future. So there's conversations around consent and assent as well. Now, this is a chart that I developed in the last 2 months with colleagues because this does not exist anywhere, which is the dichotomy of sickle cell disease care versus sickle cell survivorship care. For the most part, when we think about sickle cell disease, we think about the physical presentation of sickle cell, the social effects, everything from anxiety and depression or preventative care, the standard of care treatments available, and wanting to try other products on the market. But hardly do we talk about the impacts of these treatments on the patient's physical health, psychosocial health, but also surveillance. Currently, for transplant, the study criteria is 5 years. They will follow you for 5 years, and after that, you're on your own. For gene therapy, because it's a novel treatment, it's 15 years. But let's be honest. I know a patient who's 20 years post-treatment and just had a second hip replacement. In the conversation, it wasn't shared that there might still be a likelihood of requiring a hip replacement 20 years after transplant. So really, we need to think about what are some of the effects of cell and gene-based therapies when it comes to everything from conditioning, but also a change in the patient's like GI tract or a presentation of new and other symptoms. How are we doing longitudinal studies to look at patient-reported outcomes that aren't necessarily documented in clinical settings, but the patient's experience while they're at home? In my case, I live in Seattle, Washington. I had my transplant in Bethesda, Maryland. And so I'm always playing a game of tag, you're it, with my providers, where the transplant specialists are in a different state, and my home specialists, they aren't experts at cell and gene-based therapy. I am new and exciting, but they still don't know what to do with me. And so it's always a conversation, and the onus has now sat on me as a patient to have to navigate care on the other side of these treatments. So this is what I think the decision ecosystem needs to look like, because this feels a little bit more realistic to what patients are experiencing today. The first is thinking about the clinical factors. Number one being disease severity. If your sickle cell diagnosis is not complex enough for cell and gene-based therapies to be a viable option for you, you're simply not even engaging in conversations about these new treatments. We also need to factor in psychosocial factors. For example, I shared with you all before that there have been some mental health challenges, and I would like to think I'm a very well-adjusted woman. However, there have been moments where in my gratitude for receiving a transplant, there is also grief, because pain is still a part of my lived experience. And when my parents assume I no longer need medications, but I know differently, I have to cope with not wanting to tell them that pain is still going to be a part of my future. And so I think on the psychosocial terms, there are so many, it's an ecosystem in itself of things that we have to consider as we support patients in this decision-making process. Then I mentioned earlier the structural factors, but one thing that I didn't highlight is for you to meet the eligibility criteria for cell and gene therapy for sickle cell, your caregiver has to leave work for 6 months, which means there will be no pay coming in from that caregiver's income, because this process is very difficult on the body, and at some point the patient will need external assistance from loved ones. So we really need to critically think about, what does this cost for not just patients, but their entire familial unit? And last but certainly not least, my favorite part, which are the temporal factors, which is looking at treatment in a continuum, not just as a one-stop shop approach. Really considering what patients are having to navigate before, during, and after they receive these treatments. Now, to sum up this, I didn't want to come and tell you guys everything about navigating cell and gene-based therapies without a final call to action. And like everyone that has presented today mentioned, we need to have a more collaborative approach to the scientific and research space. For researchers, I think it's so important to really think about patient readiness. It's not just a conversation of like, does this patient want to receive gene therapy? It's critically thinking about, are they ready? Do they have enough knowledge to critically understand what they are about to sign up for? The second thing is for clinicians is really to start thinking about trust and recognizing that because this specific patient population doesn't trust the system, at every point in the informed consent process, there needs to be reminders that this patient's care is going to be centered. We say this a lot in the sickle cell community: you cannot build something for us without us. But in cell and gene therapy for sickle cell disease, it's critically apparent that we were left at those conversation tables because things were developed, and now as patient advocates and as a patient advocacy leader, we're having to critically fill those gaps that were missed. Now, for industry partners, you need to really align with patient support programs. One of the things we're developing at Sickle Cell Prodigy is peer-to-peer support, where patients who are 2 years post cell and gene therapy act as mentors for patients that are interested in embarking on this treatment journey, because again, like I mentioned, sickle cell patients are like fingerprints. We're all uniquely different. And sometimes having someone who has walked that journey with you makes the journey less lonely and less burdensome. And lastly, for community organizations, we have something everyone wants, and that's trust. People trust us because we are in the community, we are for the community, and we will continue to stand by the community. And so it's critically leading this navigation process for patients and their families, because we know that we are the bridge that industry is trying to get access to, clinicians want access to, and so do researchers. What needs to change? I think for my presentation today, this kind of feels redundant, but I will share it. Co-designed decision aids, because in the decision-making process, we need to critically decipher between how to help pediatric patients get to a place of understanding and knowledge where they have some idea of what they are embarking on, but also helping caregivers to better support both pediatric and adult patients. And so my hope is that every authorized treatment center will have some co-designed decision-making aid where patients can feel seen in the way this conversation is being conveyed. The second thing is longitudinal support models. We need to follow patients not just when they are in the clinical setting, because oftentimes, and I always say this, as people, you might not have a chronic illness, but at some point we will all be patients. You're gonna need that annual physical. So we're all a patient. And we need to critically think that sometimes patient symptoms don't always show up when they are at the appointment. Oftentimes, we all are dealing with symptoms that occur on a day-to-day regular basis. And so there needs to be a way to track patient symptoms so that we can set realistic data points of what life on the other side of these curative treatments look like. Third is survivorship infrastructure. It needs to no longer be an afterthought. It needs to be incorporated into patient care models. And I think this is critical because, yes, sickle cell disease now has gene therapy products available to us. But the reality and the hope is that other rare disorders will have gene therapy products available to them as well. And so if you get it right with us, then you run a lesser risk in getting it right with another patient population. And last but certainly not least is community-informed engagement, which is really looking at patient advocacy groups and nonprofits as key stakeholders, not just participants. And I think this is for everyone in this room, in your own small way, in the work that you do. If it means using me as the face of other patients that you are taking care of, then let that be so. But I really do think that when we design things for patients, we need to critically put our minds in their shoes and walk their walk to be able to understand how to best create infrastructure that supports them, but also their ongoing journey with the healthcare system. Thank you.
Thank you, Umi. I'd like to invite the presenters back up here to the panel, along with two other panelists. Mr. Ryan Fisher is the Chief Operating Officer at the Foundation for Angelman Syndrome Therapeutics, or FAST, F-A-S-T, and Dr. Suad Mesahel, who's the Chief Operating Officer at Aurelix Bio. And just like before, any immediate questions, comments from the audience, feel free to go up to the microphone and ask your question. I think to start out, though, thinking about patients, family members, because it's a whole group of people that are impacted by getting a cell or gene therapy in a clinical trial setting. At like the individual level, and then almost at a larger level, understanding what going into a phase 1 clinical trial means, or first in human clinical trial. I sometimes think about or worry about that, how we're communicating what that means and what the purpose and objective of a first in human trial is, especially in populations where there aren't really other options other than supportive care. How we're communicating that with families, and striking this balance between understanding what the research question is being asked and feeling like I'm getting a treatment for myself. And so I'm wondering, initially, maybe Scott, Suad, others, your experience thinking about how you talk to families about the goals of a first-in-human trial and what that means for them as individuals and the broader community.
So an easy question.
What's a phase 1, 2?
What's a phase 1?
The concept of informed consent has already been challenged a little bit, and I think we have to be honest about that. I've been in that situation and provided informed consent around first in human on multiple occasions. It's difficult. You can say all the right things, but that doesn't mean that individual is hearing all of those things. And at the end of the day, my experience is that they've usually already made up their mind by the time you're in the position where you're talking about informed consent for something that's going to happen in the near future. I don't know the answer to it. I don't, we're doing all the right things by the book, but families are desperate. They're looking for an option. The people who are coming forward to you at that stage for a first in human have a risk tolerance that is accepting of all the terrible things you're telling them might happen. And so I think in reality, it can be very difficult to kind of think through that.
Ryan, I know we've talked about this a bit, but the concept of phase 1, 2 experiment versus seeing the phase 1, 2 as a therapeutic opportunity. I don't know if you can
Yeah,
speak
I
a
mean,
little to that.
I think part of the problem is branding. Gene therapy has therapy in the name. And I think that balancing hope and expectation can be really difficult for families. And then you imagine a catastrophic diagnosis at a young age, a family going through so much through that diagnosis and then being presented with this. I think we have to be really careful with how we speak about this with them, because some just want to sign that consent and they want out. And it's understandable. But I think we need to do more for mental health support and community engagement as well, and education around this, because we have to arm families and patients with the right questions to ask in those meetings. So I think those meetings are a blur, right, for many of them.
I also want to add that obviously a phase 1 trial, there's a huge misconception that there's a treatment available, when really we're just at the very beginning of testing a treatment, and really with the priority being evaluating safety predominantly. And that, you know, families who are navigating the complexity of a rare disease are now, you add the layer of the complexity of participation in the clinical trial, it just gets even more complicated. And really, you know, providing the correct information on how complicated this is. Sometimes they need to travel from, you know, long distances to come to the clinical site. They're away from home for a long period of time. And so all of these things are really should be communicated accurately when prior to participation in a clinical trial.
I think it goes even further than maybe the first in human phase 1 or even phase 2. We have FDA approved products now that we still have a tremendous amount we don't understand about them, particularly long-term impacts. We're not even doing a good job communicating this effectively in the case of an FDA approved therapy. And in some cases, the answer in a phase 1, phase 2 is, well, we don't know. So you're just like, how do you cancel all the things that you don't know? Well, I don't know what's going to happen. Great, that's right out of the mind. Like, I don't have to worry about that. As opposed to, these are all the things that are going to happen or could happen. What does life look like after, right? We should be better about it, but there's so much we don't know that we tend to not have those as well as we should, those conversations.
Can I add something? I think I'm trying to phrase this in the nice way possible, but I really think the informed consent process needs to happen in 3 cycles. The first time has to be with the patient, where you simplify the language, right? Because I think for patients as well, walking into that room is very intimidating, because we want hope, but we also are nervous that we're going to have so many medical terms used that's going to fly over our head. So I think the first conversation needs to happen with the patient. Then the second conversation needs to happen with a patient and at least 2 members of their support family, whether that be a parent or a spouse or a sibling. Because oftentimes when you have 2 people in a room, both people are hearing different things. And so when they circle back as a unit to go home and discuss it and flesh things out, then there are certain things that patient would have picked up that they would have not picked up the first conversation. And then the third, which is to me is like the final round, is it's so much more than just picking up a pen and signing paper. This is my transplant was the most complex and hardest decision I've ever made in my entire life. And so I think that final round is now where I, as the patient, I've done my due diligence of consulting with my social, community, and then I'm coming with you as a provider and saying, if I put pen to paper, can I trust you as a provider and your team to carry me through the finish line? I think if we're doing things, it's also that, again, we critically think about conversations around like marriage and, people do pro and cons lists for like what college they're going to pick and go to. And I really do think the informed consent process, it cannot be a one-stop shop, because at every point in time, the patient has new concerns, but also their decision-making process has now been filled with an ecology of other so-called experts who are chiming in and giving feedback as well. I think if we critically think about it from that perspective, then maybe our approach to informed consent wouldn't be one and done. And then for the patient who's signing, it wouldn't be so scary, even though they want this cure. You've at least helped to level set the playing field for them.
Yeah. I mean, are we preparing the clinicians enough for this process as well? That's one thing that I worry about. So I was in Duchenne prior to Angelman syndrome for 18 years. And one of the projects we did around gene therapy was to interview 30 clinicians. And we asked them how they would describe gene therapy to their patients who might want to participate in a clinical trial. And there were a lot of varying degrees of response about benefit and risk. And it was quite frightening. So I think there is a piece that we need to do in terms of, are we training clinicians enough around informed consent and process?
I think your comment about phase is critical. I think the shift happened maybe 3 years ago. I went from cell and gene therapy, this is a distant future, and not having in-depth conversations to, I never have a clinical visit now where I don't talk about cell and gene therapies. Even for disorders that do not currently have anything in clinical trial, that there's something on the future in the horizon. But I think that's important because you're having those conversations along the way. You're giving family additional information that's allowing them to think through what makes sense in my situation, what are these risks, and it's not one conversation. But from a regulatory perspective, it all boils down to a documentation that happens at one point in time, and that's probably overly simplified, right? If we're doing it well, it'd be nice if we were actually documenting the way in which there's been numerous conversations that have led up to that point.
This is amazing. I feel like I'm learning so much on this panel too. So Wumi, you talked a lot about survivorship concept, and so I've always thought of cure as a four-letter word that we shouldn't use because it means something different to every human that's using that word, and I think it's unfair to assume that when I say cure, you hear me say cure the same way.
Right.
So, but I also love this idea that we're not fixing a disorder, we're changing the trajectory of the disorder. So I'm curious if there's anything else you could expand on, and just how can clinicians, scientists, I'm thinking about like when patient advocacy groups are meeting with Dr. Bradbury for the first time and thinking about what could that future look like. Are there any other thoughts you could add to that?
Yeah, after my transplant, my life is transformed. That's just a simple assessment, right? I think when we use because language is so important, and I always say this, is like, it's not a one-size-fits-all. Survivorship is gonna look different for a woman than it is for a man. So I think when we're having these conversations, let's really think about palliative care, all facets of a patient's life. Because I wish this was a singular decision, right? Where I'm just kind of like, I have sickle cell disease, I'm gonna get this transplant, and then pain is gonna go away. That's not my lived experience, nor my reality, right? I need to think about the impact on my mental health. I need to think about the impact on the change in family dynamics. I have 2 survivors who have now had divorces because in their marriage, they were the needy partner, and that dynamic worked. And then now that they're cured, they want to conquer the stars, and their partner is like, I didn't sign up for this version of you, right? And so realistically speaking, like, there are so many facets to a patient's life and so many areas of a patient's life that require care as well, that even though for a disease like sickle cell where it manifests and they're like physical symptoms, there are other less discussed areas of a patient's life that are equally impacted as well. I have family members who have not spoken to me since transplant because they weren't supportive of it in the first place, right? I have other patient advocates who challenge me and say things like, well, how can you say you went through this treatment process, but when you have children, your children will still have the sickle cell gene? But that's my reality, because when I froze my eggs, I had sickle cell disease. So I'm still gonna pass that genetically to my offspring. But because we aren't thinking about, it's only now that conversations around fertility preservations are even happening. When I did my transplant, I was the very first NIH patient that they covered fertility preservation for. And I made sure that after me, everyone else would get it covered. Because it shouldn't be an afterthought when we're thinking, like, fertility is such a poignant conversation. A lot of relationships are built on, I want children, or I don't want children. And so I think if we think about this from a holistic point of view, then it will change how we have these conversations, because, you know, there's something that was very apparent to me after transplant was I started to question everything about the way my disease was presenting itself, to the point where for some months, I thought I was losing my mind. Because the reality was, no one prepared me for short-term memory loss. I am very good when it comes to like random facts, and then all of a sudden I was like struggling, and you know, for other colleagues of mine who were getting their PhD, getting their master's, they had to withdraw because they weren't prepared for the, you know what I mean, like the psychological, mental health issues that were going to, you know, happen at that point in their recovery as well. So I really do think, like, when we think about these treatment options, let's think about it not just from a clinical endpoint of, I'm going to see how you're doing 6 months from now, 12 months from now, 18 months from now, and checking how the disease presents itself, but also paying attention to like these psychosocial aspects of things, but also the reproductive aspects of a patient's life as well.
I just want to add also that, you know, we definitely want to move away from the word cure, as we are still evaluating the product. We have absolutely no idea whether it is even safe or that it's going to change outcomes for patients. And so we have to be very careful when we addressing the patient community, how we're actually, you know, pitching the clinical trial or even the product, and that typically if the product is available now in clinical trials, we're really not even in the right patient population that it's really going to make the difference in, because current patients who are enrolled in these trials are already symptomatic. They are likely not going to really benefit from the full you know, treatment of this product, but these are the patients who have you know, currently available to us to try the products on. So it's a really difficult position to be in when you really do need the patient community to step up and be the first. But also what we know is that it's the ones who aren't even born yet are the ones who will truly benefit from the product if it shows obviously safety and efficacy.
Yeah.
So. Yeah.
I agree. I think therapeutic misconception happens a lot in rare disease in particular, but we see it even researchers and companies, and so it's a responsibility, I think, across the ecosystem of how we talk about these things responsibly, because families are hanging on every word. I mean, I've had families call into investor calls to hear what's going on, not realizing the sort of tone of that is a little different than what you might get at a conference. So words do matter, and I think it is something that we need to really look at.
Yeah. So kind of in this vein of the landscape is changing, right? And I think Ryan, Swad, you guys both represent people that are kind of on the vanguard of change, right? Like you're patient advocacy groups that are doing very unique, amazing things with FAST, but you also have your DMD experience from the past. And Swad, you've been in really cool companies that are doing really cool, new, innovative ways of like solving these problems so that we can actually pull forward some of these learnings and not just do it the way we used to. I wonder if y'all could talk a little bit about like paving a new path, going in a different direction.
Yeah, I mean, you know, part of it is really creating this consortium of understanding that we can't do this alone, for sure. Industry can't do this alone. The patient advocacy organizations can't do this alone, and neither can the clinicians. And so we really do need to create very you know, strategic partnerships where each entity is bringing something extremely valuable to the table in the drug development process. And it's really not a transactional relationship. This is a true partnership where we truly align on, at the very beginning, on what the goals are and the needs. Because there's the wants and then there's the needs. And if we're all aligned on the needs, that really helps, you know, the clinical development and the long-term partnership, you know, and sustainability really, to be able to support the rare disease community. But there are new models that we're always trying to be creative and think, what else can we do to solve this problem? And we've got now non-profit biotechs emerging. We have, co-development partnerships are developing, in the space. So we're really trying to be innovative, to think what else can we do to solve the rare disease crisis, really, where unfortunately with rare and ultra-rare diseases, industry partners aren't really coming to save the day, and it has to be done with a new model, a new partnership, a new consortium, really bringing all of the different entities together and do co-development as a team.
Yeah, so FAST was started by a group of parents who have loved ones with Angelman syndrome, and we're a venture nonprofit, I guess I would describe us as. But the system, the paradigm was essentially failing our families in terms of how nonprofits fund research. You know, you sort of maybe pick a few researchers, bet on a few horses, and hope for the best. I think we were lucky enough to have parents who had deep expertise in drug development and translational research, where we created a plan. We sort of brought together researchers from institutions across the country to make them collaborate, force them to collaborate. And we ended up funding early stage research for 6 different strategies, hoping that one would bear fruit. And one of the big things, and Scott talked about this, Allison touched on it, our model was to go after IP. And if necessary, start a company to take this forward beyond the early stages and into IND enabling, and then if necessary, if we could, fund phase 1, 2, and get some data to de-risk the process. At the same time, in parallel, we were setting up the infrastructure, all the things you heard about, natural history studies, validating outcome measures, characterizing the disease, understanding what meaningful benefit looks like, and engaging the patient population rigorously, and then bringing together this sort of ecosystem of players into a consortium to look at outcome measures and biomarkers. We had to do all these things together. We were lucky enough to have funding to do the early research, and then once we were able to get enough data on the one that bore fruit, which was an ASO therapy, it was eventually partnered with Ultragenyx, and FAST received funding to then take forward other strategies. And that's been our model ever since. We do go after IP because we want as much control as possible, because decisions by companies or the market or reasons things could be shelved, we don't want good science to not move forward. So we want to do everything we can to enable these potential therapies.
And I'm just going to follow up with that and just say that there are lessons learned definitely in the process, and what not to do, and really how now to target efficiencies really early on in the clinical development process, so that the resources that can stretch out and go way beyond and further than just getting to a phase 1 trial. And so it's really important to really align with industry partners who are like-minded, who really want to be able to solve this problem, who have the right expertise in drug development and clinical strategy and regulatory affairs, to know how to navigate the regulators and what they're really looking for, and to get a single trial from, before you've even treated your first patient, you have this alignment with the regulators that you can go from, you're able to scale up and get to the end much quicker, because you've targeted these efficiencies really early on in your clinical development process. And that is what we're now learning, how to do that better and how to do that right. And that's really what we do at Oralex, is bring patient advocacy groups really early on in the process and streamline that process so that you can get to your goal, which is really approval, as quick and efficient as possible.
Scott, I wonder, so I caught something that sounded like an analogy to me. So when Allison was up here earlier talking about the preclinical side, you're thinking about risk, time, money, and all the strategic decisions, and this kind of ties into what Suad's talking about of how you're making your overarching plan I wonder if you could talk a little bit more about thinking about your clinical trial readiness, your phase 1-2 versus phase 1-2-3 planning, and kind of how does that all tie in here too?
Yeah, I mean, I think a lot of this has to do with the characteristics of the disease population you're dealing with, right? So are you talking about something that's rare, but like rare, like there's several hundred patients in the country? The strategy that's going to work and be acceptable to the regulatory authorities is going to be different than an ultra rare where there's less than 100 or less than 50 patients known in the United States or in the world. What's realistic and what's possible around aspects of clinical trial readiness, and also to what extent is this a disorder that is similar to other things that you can lean on from a standpoint of endpoints and likelihood that they're going to translate effectively. Or this really is quite unique and nobody's done any work around good endpoints in similar disorders, so you can't lean on that, right? So you have to kind of assess what are the critical gaps across the laundry list of things that you want in place to be able to move a program forward, and decide how do those resources get best allocated at an early phase. I don't know how to give it more specifically. It's different for every situation.
Yeah,
That's what you guys are going to help them do.
I
right?
think that's the important thing, is that it's not a one-size-fits-all, but look and see what makes sense for your group based off of what others have done, and then do you push the envelope in other places? Yeah. And as a reminder, the mics are open, so if y'all have any questions too, we don't have to eat up all the oxygen in the room. But I wanted to get back maybe to Wumi as we're thinking about that, you know, the tension between approved products versus clinical trial products, and you know, now you're at the almost like the back end of things, trying to get the industry to catch up to what they should have done earlier. Is there any kind of watch outs or things you could share with?
I think I'm very lucky that I'm in a position where the companies that have products in the market I've worked with for a while. And so there's been some changes happening. They're not happening as quickly as we'd like. But, you know, as long as we're heading in the right direction, that's what's important. But I think one other thing that's so important is, as a, you know, biotech pharmaceutical company, is, you know, when you are developing these clinical trials and products, always pause to ask yourself the question of, is this an avenue where we could leverage a community voice in that conversation? Because I really do think that, like, what I'm seeing in sickle cell disease in particular is we're all working in silos. And so what's happening is, like, some people are doing really, really good work, but because there's no cross collaboration, very few people can get that benefit. But I'm happy to see that, especially with the, you know, I have to salute industry partners, because the companies I've been privileged to consult for or work with are doing the hard task of making sure that we are looped in everything from simple things like materials being used and distributed among patients. And so it feels inclusive for us as a patient community, but also it serves them better, because in the education process, they're starting to see where there's been an uptick of interest, even in families that their primary language is in English. Right? And I really think, like, in developing these treatments, you really need to think about things bilingually, because there are other communities that are limited in their understanding of the English language that also need resources developed for them as well. So with you know, we're in an era of AI. We have a partnership with an organization that's developing AI tool for sickle cell that's like a ChatGPT for sickle cell disease. And we've had to do it in 4 languages, because it's so important for us to be very cognizant of the Arabic community, the French-speaking community, and the Spanish community as well.
No, that's great. We need more, like, butterflies cross-pollinating all these different spaces. Awesome.
Yeah, I wanted to ask about, so what really resonated with me was the whole discussion about informed consent, and how it's really hard to communicate what people are consenting to and just the fact that it's even a fluid target. And then, but layered on top of that, we now, especially in cell and gene therapy, there are a lot of accelerated approval products, which have the even less evidence supporting their use, but have the appearance of FDA approval, right? So, you know, and I don't know that how many people can distinguish between accelerated approval and approval, you know, full approval, you know, to the degree that that means something. So how do you, what are your thoughts on communicating sort of risk levels to patients? Because now you're dealing with something that, well, could work, but we don't really fully understand it. The evidence base is incomplete, and it's in this weird regulatory position that people aren't used to.
Do you want to go? I was just going to add, it all depends on the disease.
Yeah.
Because if it's a fatal disease and families have got no other alternative, they are more likely to try an experimental therapy with the risks associated with it than a patient who is likely to live to adulthood and you know, just have a more improved quality of life. So it really depends on the disease.
Yeah, I mean, and I think that there's an element of tolerance for uncertainty. So I think we really need to be clear about how much evidence and information we have about these products. And if it's an accelerated approval, what does that mean? Well, was there a biomarker? And that states it's reasonably likely to predict benefit. But I think we need to dig into what exactly that means. We owe that to the patient community so that they can make the most informed decision at the end of the day.
I think it's critically important to have expedited approval pathways for rare disease, but we also have to be very honest about what that means. And I think one of the things that's been a little bit challenging is what does post-approval look like in terms of the rigor of monitoring ongoing information, dissemination of that information. I'm actually very much in support of leaning on the direction of I want to increase access, particularly for things where there is no alternative treatment. But part of the ability to do that responsibly as a clinician is to have as much information as up to date as possible as we continue to learn about it and are continuing to think about survivorship and what comes after, and long-term follow-up and survivorship kind of come together, and we need to be able to inform that in an ongoing fashion, even for individuals who have already received a treatment and are continuing to follow with you, so you can continue to update what is my surveillance, what is the things I'm monitoring for, how am I talking to patients, even if they've already been treated. And I think there's a little bit of a like, great, it's approved, fantastic, we'll let you know if something terrible happens, right? and that's probably not good enough, and we should be demanding rigor in our phase 4s and post-marketing as we are in the pre-approval scenarios.
Thank you.
Scott, you alluded to plausible mechanism, and you might want to talk about it on the panel.
I don't know that I want to talk about it.
I don't know if anybody wants to kind of touch on it, because it is the newest hot topic coming out of this FDA.
Yes, Scott, please.
I could offer nothing other than my own interpretation of the document.
That's all we can all do right now.
Which I mean, nobody's tested it, right? So we don't know what it means, but it attempts to lay out a framework that allows us to move existing statute around how you can demonstrate safety and efficacy to more and more rare populations. That's essentially what it's attempting to outline. We could get into all kinds of nitty-gritty, which I don't mind nerding out about because I enjoy it and find it interesting. But part of what I would say is one of my biggest takeaways from it is rigor. Yeah. Is that it in no way shifts the onus of proof off of the investigators, companies, sponsors from demonstrating safety and efficacy. Right. And so the only way you can do that with a smaller and smaller population is through really careful planning and highly rigorous data that is very comprehensive upfront, whether that's included in natural history and cohort design as an external control or with patients as their own control as a lead-in, which is, in the N of 1 space is the only option, right, as the most extreme version of an ultra-rare disease. And so I think we have to kind of think about how we're doing development programs to bring that level of planning and rigor up front. And I always like to plug, and ideally not reinvent the wheel every time. So how can we sort of rationally cohort similar diseases together to allow scaling across rare diseases for efficiency of resource utilization, funding, et cetera, in ways that make rational clinical and scientific sense.
I think I'll say this one comment, we'll get to that last question, but it's when I was in academia, I thought a lot about the what are we going to measure, and now that I'm on the industry side, I think much more about the how are we measuring it. So I do think both of those questions are critically important. Okay, last question.
Wumi, Dr. Demers, thank you for your insightful talks. Brian Moy, Jaguar Gene Therapy. Just I asked the panel to talk a little bit more about like disease-specific CGIs and outcome assessments and kind of the PAG's role in developing them and kind of that cross-interaction with industry and kind of validating them for rare diseases. Thanks.
Yeah, I can start. The Angelman syndrome community actually did a patient listening session a number of years back because we felt that the current measures were not sufficient to capturing what was most meaningful and the nuance, as Scott was talking about in his presentation, of that sort of smallest meaningful benefit over potential risk. And the listening session led to the development of a caregiver-reported outcome called the ORCA. This was 2015. Here we are over 10 years later. It's still not a primary endpoint. It's in all the trials, but what I will say is that, and there's been a lot of work, it's been validated in other NDDs, other conditions. I will say that it takes a long time for these things to mature and finally get to the point where it might be considered a primary endpoint in our experience.
Yeah, how do you know you have a really good outcome measure? It's been used as a primary endpoint for an approval. So short of that, how do you jump to get to that point, right? It's a bit of a catch-22 of the requirements to get there. I think when it comes to global measures and CGIs, it's worth, maybe I'll just do 30 seconds on my soapbox. Are you doing a global impression, and there could be of severity or of change, as an orthogonal validation of another existing endpoint, which says this is also meaningful and directionally similar to some other endpoint? Or are you trying to generate a new outcome measure, scale, for example, like ORCA, or other ones that are meant to be the endpoint? Yeah. And there is often not, we use the terms interchangeably in a way that gets a little bit distracting from being able to have clarity of, is this meant to be the endpoint or is it meant to be a supporter to the endpoint? And we want to be careful because the requirements of each of those are very different.
Yeah, and just I want to add that the FDA really pushes drug developers to look at more binary endpoints, which is achieved, not achieved, survived, not survived, and very objective endpoints where there is very clear delineation between the treated population and the untreated population. And obviously it's very difficult to do, especially in the neurogenetic space where some of these diseases take slow progressing diseases, for example, and how do you measure a treatment effect? And so that's, I think, is the hardest bit when we start throwing in scales that haven't been validated and putting that in front of the FDA and saying, well, there's a 2-point change on this scale, and will those say, well, how does that translate to something clinically meaningful? Did the patient walk faster? Did the patient, is the gait improved? And so you really need to be able to translate what's in the scales to something clinically meaningful. They won't just take the scales. That's even the biggest problem, that you go to all of this effort to develop your disease rating scales, and that's also a challenge to really get them to align on that as a primary endpoint. So it's a huge challenge developing and aligning outcome measures and endpoints with the FDA, especially in the neurogenetic space.
Mm-hmm. Yeah, and there's new guidances for PFDD around these questions. However, many of us are doing what you were talking about. We have measures that are used in neurotypical children and sort of validating them in our conditions, and they have limitations. They're not perfect. But we are able to still put them in trials, and we're hoping for the best. We have a few phase 3 trials. We'll let you know.
I think that's one of the values you can get out of a natural history study, if you're lucky, is to try to identify certain events that either never happen or very rarely happen. And if that gives you that binary. So you may have an entire scale, but you can say these things just didn't happen in this population in a natural occurrence. That is one of the things the FDA seems more open to accepting as a binary outcome that you can compare with external controls, compared to they had a 2-point improvement and what the heck does that mean? And maybe more liable to have fluctuation associated with it.
This was incredible. We're at time.
We are at time. Thank you everyone for joining today. Thank you again to our panelists and presenters throughout the workshop. And enjoy the rest of your conference.
Off to the airport.